Hello students, welcome to Vedantu NEET English. I'm Ms. Gopika, your own biology master teacher. I hope all of you are ready for your backlog series, and today we are going to do locomotion and movement. Last class we have done animal kingdom, and in the previous class also we have taken up human physiology chapters like your excretion, body fluids and circulation. So do not fail to see all the lectures, as now the NEET pattern has changed. Basically section B has been removed and they are also going to go for a pre-COVID era. So in the pre-COVID era, if you see the papers of 2018-19, you will notice that the questions in biology are more conceptual. They ask you to think, okay, not just by heart and write. So this time competition, all that you can leave it aside, but the real hard work is going to be paid off. So I want all of you to work more hard in biology. Don't take it very easily, telling that, oh, it's biology, it's so easy. That will not work for you to get 360 out of 360. Okay, yes. So let me just refresh your chats, okay, because I'm not able to see any of your messages. Yes, now I can see. Yes, students. Let the energy be high. Okay, if your energy is high only, other people also will feel like, okay, they have to study. Okay, yes, I'm doing great. I hope all of you are also doing great, right? Yes, shall we start? Okay, locomotion and movement. Again, as I always say, all the human physiology chapters have a weightage of maximum of 3 questions. So that means this chapter also will have the same thing. Around 12 marks you can expect from this chapter. But let me tell you, this 12 marks is very easy to gain, because locomotion and movement is not very difficult chapter. And I've also made tricks for you to be, for you to make it very easier to study all the names. Okay, yes. Oh, nice. Okay, nice. Yes, all of you quickly smash the like button and subscribe to the channel if you haven't, because lot of NEET content is going to be coming and you are going to be bombarded with lot of, your pre-COVID pattern questions and practice questions. We have planned so much for you. For us to do all of that, I want all of you to be more cooperative and more, attentive in class. Okay, yes, let's start, students. Locomotion and movement. So don't worry about the session link. You will be getting the session PDF in the description box. You will also get it on the Telegram channel Biology by Gopika and Vedantu NEET English Telegram channel. Vedantu NEET English Telegram channel, you will get all the notes. PCB's notes you will be getting. Okay, yes, so don't have to search the notes anywhere and all that. Okay, all that is not required. Now students, the chapter speaks about locomotion and movement, right? So what is this locomotion and movement? Now movement means what? Like this. All of you stretch your hands, okay, up, down. Okay, they'll be thinking it's an aerobic class. Now what am I doing? I'm doing a movement, but now I'm moving like this, I'm moving this way. This is a locomotion. That means I'm changing my location. I'm moving from one place to another. That is called a locomotion. And movement means you do not have to move anywhere. You do not have to change your location, but still you are making slight movements. Okay, slight movement. So here also remember, locomotion means moving away from the original position of an organism. It's called the locomotion. That means what, students? Here there is a displacement that is happening, or there is a change in location, right? Change in location. Agreed or not? Did all of you raise your hands? Yes, all of you raise your hands in the chats also. That is your movement that you're going to do. Okay, then students, here if you talk about, when I speak about movement, movement can happen without moving from one location to another. See, I stood here and I did this. See, now, does that mean I'm moving from one place to another? No, but I'm moving my arms. Okay, now let's say I am moving my, eyebrows. Okay, like that. Okay, you ask each other, no? Which question? 1, 2, 3, 4, like this full, Bharatanatyam you'll be doing in the exam hall, no? That movements, okay? Tongue say we do movement, jaw we can do movement. So all these also can happen without have to change the location, right? Without changing the position, I can do something. Okay, that is called a movement. So movement can happen with or without moving away from an organism original positions. Okay, it is always voluntary. It is neither voluntary or involuntary. Clear or not, all of you? Yes or no? Perfect. Now, one of the assertion reason question can come from the statement, students. What is this assertion reason question? All locomotions are movements, but all movements are not locomotions, right? It's kind of like a tongue twister, but there's nothing to be worried about, because here understands all locomotions are movement. That means, see, now without doing a movement. Okay, all locomotions are movement, but all movements are not locomotion, because when I'm doing this, I am basically not shifting my location, I'm not moving from the original position. So how can I call it locomotion, right? So here all locomotions are movement, but all movements are locomotion can come as an assertion reason. So I want all of you to highlight it in your NCERT. Okay, done. This is basic introduction of it. Now comes the actual chapter. Students, there are types of movements, and you'll be like, ma'am, types of movements? Yes, there are so many things that happens in our body. Many of our cells are doing different kinds of movement. In that, you have to mainly study 3 movements: amoeboid movement, ciliary movement, muscular movement. Then I will also tell you flagellated movement. Okay, ready all of you? I want all of you to write lecture notes. Okay, I will share these notes on my Telegram, but still write the lecture notes, and on the description box also I will be sharing this. Now students, amoeboid movement means what? Amoeba moves with the help of what? Tell me in the chats. Okay, this is your amoeba. Amoeba moves with the help of pseudopodia, right? Amoeba moves with the help of pseudopodia. So pseudopodia is helping. Yes, ma'am. But I am telling you that in our body also there are some cells that are doing amoeboid movement. Yes, very good, very good. I can see some of you answering. Which are the cells that are doing this? The cells are going to be the ones that are helping you with the immune system. That is your WBC or your leukocyte and your macrophages. Macrophages are what, students? They are the factory of your HIV cells, right? HIV cells come into the macrophages, multiply and make them as a factory. So WBCs and macrophage cells, and also some of your cytoskeletons, okay, in your body will be able to do this amoeboid movement. Now exactly how do WBC do the amoeboid movement? Recently only I taught you in your body fluids and circulation that through the capillaries there will be tiny pores, okay, through which this WBCs will squeeze out. Okay, this WBCs will squeeze out, and this is called as what? All of you tell me in the chat box. All of you tell me in the chat box. WBCs will squeeze out through the small, okay, pores, which is called as diapedicles. Very good. Okay, diapedicles. So that is how WBCs, and because of that irregular shape, they'll be able to squeeze out of that small pores just for what? So that they can protect someone else. They see that, oh my God, there is something happening there. They will be like, I need to protect them. So they will squeeze out of this pores and they will go outside. This process is called as diapedicles. Okay, clear or not? This we have learned in our body fluids and circulation. Body fluids and circulation, we studied this, so you people will be able to remember. Very good, very good. I can see some of you answering. Excellent. Okay. Now, next thing, what is it, students? So, amoeboid movement, you understood. We know that amoeba, with the help of pseudopodia, which is also called the false feet, they're doing the movement. Now, in our body also, WBCs, macrophages, some of the cytoskeletons, they are also able to do the amoeboid movement. Okay, now comes the ciliary movement. Ciliary movement, my students know, so many times we have studied it. Breathing and exchange also we have studied. Now, for bringing the ovum from all the way, with the help of fallopian tube also, we will be studying about the ciliary movements. Now students, where do you see the ciliary movements? Now let's take an example of an organism. If I have to take an organism, whose example will I take? I told you, sole, your shoe sole is there, no? Paramecium, right? Somewhat like this it will look. Somewhat like this it will look. Okay, so like this, cilia will be there. Okay, and remember, all of us studied a gullet here. No, here, and students, remember, they have micro and macro nucleus, right? They have micro and macro nucleus. So this is also, with the help of cilia, they will be able to do the slight movements, right? Now here, under ciliary movements, you have to learn about the lining of fallopian tube, the lining of fallopian tube, the lining of tracheae, right? When dust enters, how is it getting removed out? Okay, with the help of the linings in the tracheae as well as the bronchi. Clear? Yes or no? And also the linings in the fallopian tube is one of the reasons for this ovum to get, brought to the position, okay, wherever it has to be brought, no, the ovum has been transferred with the help of these linings, the cilia linings, which are going to help them, okay. So if I have to say, let's say students, like, let's say this is our trachea, okay, in this we'll have the ciliated cells, okay. I'm just drawing like this, but all of you know now how your ciliated cells will be there, yes or no? Yes. Good evening. Perfect. Okay, this examples you have to remember. This is where the question will come from. Now the last one, students, is called the muscular movement. Now muscular movement means what? Now the movement of our limbs, okay, movement of our jaws, all these are muscular movements. Now how many muscles are there in your body? All of you tell me in the chat box. Okay, so students, muscular movements, I can say of your limbs, jaws, okay, jaw, etc. And you have around 639 muscles. Correct? They're all helping us for some kind of movement. Now it is not necessary. Obviously, tongue also you can write. It's not necessary that all the muscles that you're studying. No, now we study cardiac muscle, we study striated muscles, or we study smooth muscles, right? So striated muscles slash different ones, smooth muscle, okay, smooth muscle or visceral muscle, striated muscle or skeletal muscle. So when we study all this, we realize that cardiac muscle and the smooth muscle, cardiac muscle which is present in your heart, and smooth muscle which is lining your inner organs, okay, those people are not voluntary. You cannot control them, but we can control one muscle, which is that muscle, your skeletal muscle. Right? Skeletal muscle, you can control full biceps, triceps, everything you can do with the help of your skeletal muscle. So is this clear to all of you? Amoeboid movement, ciliary movement, and muscular movement. Now I told you, students, one more movement I'll tell you is about the flagellated movement, okay, which you already know. Okay, flagellated movement, which my students already know. What is that? Ma'am, obviously it is the sperm. Sperm shows flagellated movement. The only thing that will show flagellated movement is your sperm. Okay, is the sperm. Okay, yes or no? Clear? Yes. Fine. Perfectly clear. Body fluids and circulation you haven't studied. Who is that? Chitra? Chitra, haven't you studied body fluids and circulation, dear? There we have studied. Human health and diseases we have studied. Yes or no? Okay. Yeah, it's just a type of WBCs. Okay, they're all going to help us with the immune system. Fine. Okay. Chalo, let's move forward. Properties of the muscle. Very, very important question. And also it is important for you to remember what you're trying to learn here, right? What you're trying to learn here. So here, students, you're going to remember this. Okay, remember this. E, C. This is how you'll remember, okay? E-E-E-C. Easy? Yes or no? E-E-E-C. Tell me, repeat after me. Yes. So, first E stands for excitability. Excitability. Okay, excitability. Now, what is excitability? Now, students, remember, your entire body works in such a way that three people have to be in sync. Who and all? Nervous system. Yes or no? Your nervous system. I'll give you an example also. Okay. Then your skeletal system. And your muscular system. These three people have to be in coordination for any movement to happen. Correct, right? Now let's take an example, okay? Karthi has touched a hot object. Now when Karthi touches a hot object, immediately the signal will be sent from your sensory to your brain telling that Karthi is touching a hot object. What will happen? The signal will come back telling that retrieve his hand, okay? Now, if nervous system is not in sync with skeletal system is not in sync with muscular, then your movement will not happen. Because if I want to retrieve, the signal has to come from the brain. My muscles, skeletal muscles should be listening to me, right? Listening to my brain. All this should happen. For all this to happen at the same time, my muscles, right? About your contractile protein also we are going to learn. They also should be ready for contraction and relaxation. So understand that these three people should be in coordination for the entire process to happen. Now, when I come back here, I'm talking about excitability, and what do I mean by that? Let's say this is your muscle. Okay, this is your muscle. This is your muscle. Now to this muscle, students, okay, to this muscle there is going to be a signal, okay, that is going to come. Okay, one signal is going to be sent. Now the muscle should be able to get excited, respond. So that is called as excitability. Clear or not? Excitability. Now the next E stands for what? Extensibility. Extensibility. The ability of the muscle to extend, okay, without causing any damage. Now when I'm stretching, there's chances that definitely muscle will stretch, okay, but it should also come back to its original position without any wear and tear, right? That is what makes sense. So extensibility is there. Then it is the elasticity, okay, then elasticity is there. And last one is going to be what, students? Contractibility. Now elasticity means what? The ability of the muscle to come back to their original position or original length. That also is a property of the muscles. Now how much ever you stretch, okay, you will not become like that, you know, like that, what do I call that? Yeah, slime. You will not become like a slime, no? Now let's say slime is in a box, okay, you remove it, you stretch it, you will become like that? No. After stretching or so your muscle will be back to normal. So the ability of the muscle to come back to its original length. And obviously the last one is going to be none other than your contractibility, which is one of the most important things that you're going to study further as well. Contractibility. That means the ability of the muscle to contract. So obviously contractibility, elasticity, extensibility and excitability is what we are going to study continuation in this chapter. Okay. Yes, those who haven't liked, quickly like the video and share it with your friends. Now you people will tell me, ma'am, no friends. I know. Okay. I know you people will tell that. Come on, quickly like the video. These likes does not even encourage me. Okay. Yes. Good job. Okay. So this is the properties of the muscle. So is this clear, students? The ability of the muscle to come back to its original position. Contractibility means the ability of the muscle to contract. Extensibility means the ability of the muscle to extend or stretch and then still come back to its original position without any damage. Okay, no damage, that is the most important. Now remember that nervous system, skeletal system, muscular system, all of this has to be in coordination with each other. Okay, they have to be coordinated for the entire process to happen. They have to be coordinated. Done? Okay, just a very simple basic understanding. Okay. Now, types of muscles, which I have already taught you in structural organization, but still I have to teach you once again about which are the three muscles. All of you tell me in the chat box quickly. What is it? Smooth muscle, skeletal muscle, cardiac muscle, right? We have studied this. We have already studied it in structural organization. So students, first one, smooth muscles. All these notes will be available, so don't take tension, just focus on the class. Okay, smooth muscles. Now, smooth muscle means what, students? First of all, understand the diagram of the muscle. That is most important. Whenever you are trying to study biology, always use pictures. Okay, maximum pictures. So, fusiform shaped. What shaped? Fusiform shaped. Okay, that means tapering at the ends, right? Tapering at the ends. This is how they are. Yes or no? This is how they are. Correct? Smooth muscles? Very good. I can see some of you answering. Yes. Perfect. Perfect. Yes. Okay. And these people are uninucleated. You can see one nucleus. Okay. So students, what is the most important characteristics of them? First is going to be the shape. You're going to remember the shape. They are fusiform, or else you can use the word tapering. Okay. Tapering. Can you see tapering? Yes, ma'am. I can see tapering. The tip. Okay. Both the tips are tapered, right? Yes. The next thing is what, students? Do you see any branches? No, ma'am. They are unbranched. They are unbranched. And where are these people going to lie? Okay, where are these people going to lie? Always remember, your smooth muscle can also be called as visceral muscle because of the main reason that they are going to be lining your visceral organs. Visceral organs means what? Internal organs. Can you actually control that? It's not possible. So here, this person is completely involuntary. Okay, completely Involuntary. So just remember their shape. Are they branched? Are they involuntary? And the next most important thing, their striations. Do they have dark and light bands? Do they have dark and light bands? Okay, I will show you some videos like this also. Students, I try to see the 3D one, but I think this board, it does not work. Okay, so I'll try to share it on Telegram. Okay, involuntary. Okay, involuntary. And students, unbranched, involuntary, they're not striped. Okay, not striped. Not striped means understood, right? No striations, no dark light bands, all that is not there. Next one is going to be what? Next one is going to be what? Your cardiac. Okay, cardiac muscle. What do you people know about cardiac muscle? Tell me in the chats. Let me see how many of you know about cardiac muscle. Again, first preference to diagram. Okay. I think I'll just draw one, students, because too many branches might look a little difficult to draw. Okay, something like this. Okay, branching is there, that's why. Done? Okay, so here if you see, I just wanted to show the branching. Definitely the diagram is not at its best. Okay, and also one more thing I want to show you is, what is this? Ma'am, they have striations. Okay, they have striations. Okay, they have striations. Understood? Yes or no? Yes, light and dark bands are present. Very good. Someone has answered in the chat box. Now, students, let's see. These people have intercalated disc. Okay, this branching which you can see now, these people have intercalated disc. Intercalated disc. And definitely these people, they have the branching. Now, most important thing, first of all, the shape. What does it look like? Ma'am, it looks cylindrical. Right? Cylindrical. Cylindrical. What is the second thing we wrote? Branched or unbranched? All of you tell me in the chat box. Yes, they are branched. Okay? They are branched. Good. Next thing, what do you know? Voluntary, involuntary? Obviously, it's going to be involuntary. Can you control your heart muscles? You cannot do that, okay? But yes, do they have striations? Yes, ma'am, they do have striations. So we can tell striated, okay, striated or striped. Is this clear? Cardiac muscle is clear? Yes, Apsara. Clear? Perfect. Good job, good job. Many of you are new to the class. Very nice. Okay, I hope every day you people will be able to join. Okay, good. Okay, yes, striated, involuntary, branched and cylindrical. The last but not the least, who is that? What we are going to study in this entire chapter, that is going to be your skeletal muscles. Skeletal muscles. Students, skeletal muscle is the only muscle that is voluntary. That means in your control. Okay, in your control. Again, we give preference to the diagram. You can draw the normal diagram or we will just draw like this. Okay, we just draw like this. Yes, again they also have dark bands, obviously striations. Okay, like this, and they are multi-nucleated. Okay, they are multi-nucleated. This is also, this condition is called syncytium. There is a word mentioned in your NCERT if you see. Okay, yes. Done. Now, what do you know other than that? Other than that, what do you know? Yes, striated, voluntary, very good. First of all, you should write it is cylindrical in shape. Okay, it is cylindrical. Second thing you have written about the branching, they are unbranched. You cannot see any branching here. These people are unbranched. Third thing, voluntary. Okay, the only people who are voluntary, so it is very special character to remember. And striated, yes, of course they are striated, and this striations is because of the presence of your A band and I band, right? A band and I band. Got it? Yes. This is Locomotion and Movement, your 11th chapter. Okay? Yes. Syncytium, that means these people are multinucleated. Cylindrical, unbranched, voluntary, striated. Is this clear to all of you? We have studied this in structural organization as well. So it is just a, just a basic understanding of it. There's nothing like, that you have to study extra. On that note, you do not have to study anything extra. Now, I want all of you to give me next 5 minutes and only listen to me. Don't talk anything to yourself, because I'm going to teach you skeletal muscle, and this is the foundation for the entire chapter. Okay, so can we start? Yes, ma'am, we should start. Okay, now I'm going to draw some diagrams to make you understand. So students, remember, bone, to the bone the muscle will be attached. Okay, this is how we draw the bone. Okay, to the bone your muscle will be attached. Okay, this is how we draw the muscle. Yes or no? This is the muscle, correct? Now, to the bone, if the muscle is attached, it has to be attached with the help of something. Yes or no? It has to be attached with the help of something. What is this? Okay, bone to the muscle. The attachment is tendon. Okay, this is a tendon. This is an entire muscle, right? This is an entire muscle. An entire muscle. Now say students, if I draw the muscle, okay, once again, like this, okay, like this. Okay, if I draw the muscle like this, just to, just this portion I'm enlarging and drawing so that I can draw everything neatly. Okay, and let's say we just give a connection like this. Okay, just a connection like this. This is just a normal muscle, nothing big about this. Okay, now, in the muscle, there are going to be muscle bundles. Okay, this is your entire muscle. Yes, ma'am, this is my entire muscle. One second. Okay, this is my entire muscle. Okay, yes, better? Makes sense? Okay, very good. Tendons, everyone has answered. Very nice. So, what did I say? This is your entire muscle. This muscle will have so many muscle bundles. Okay, they are not going to be in regular shape. Everyone draws it circular because it's easier to show. Okay, they are going to be many muscle bundles. What are these muscle bundles called as, students? What are these muscle bundles called as? Yes, very good. Fascicles. Okay, these are the muscle bundles. Okay, muscle bundles, or called as, most important name, fascicles. Okay, got it? Fascicles. So the entire muscle is there. To the bone, the muscle is attached, called as tendon. That is called as tendon. The entire muscle has many muscle bundles called as the fascicles. Okay, fascicles. Got it? Yes, ma'am. Understood. Now, inside this, what is there? Let's just take only just one muscle. Okay. Now, inside this, there are muscle cells. Okay, inside this, there are muscle cells arranged like this. Okay, arranged like this. Like this. Okay, this way, muscle cells are arranged. I can call that muscle cells also. Lag is there? I don't see any lag. Who is this Prakash? Is there a lag, students? Or this is just a spammer? Time pass. Lag ma'am, lag ma'am. He wants to lag in his life. Okay. Yes. So, let's draw here also 1 or 2. Okay, so that I can show the entire diagram for you. Yes, these are called as muscle fibers or muscle cells. Okay, muscle fibers or muscle cells. Now, understand, this muscle cells or muscle fibers, why do I call it fiber? Because they're long and slender. So, let's just draw one big muscle, okay, muscle fiber. About the layers we will study. Okay, about the layers we will study. Yes, what did I pull out, students? I pulled out a muscle fiber. Okay, muscle fiber. Done. Hey, now you people don't tell chuma. Chuma means simply. Okay? Okay. Now look here. This is a muscle fiber. Done, ma'am. Inside this muscle fiber, there are going to be smaller portions which are called as myofibrils. Okay? Smaller portions which are myofibrils. Okay? Inside this, there is going to be myofibrils. Like this. Okay, let me just draw one. What is it? Myofibrils. Understood? One I stretched out. That is a myofibril. Got it? Now, this inside this myofibril is where you are going to study your most important actin and myosin. Okay? You will study about your myosin. Okay, myosin has come out. Can you see? Yes or no? Since if you talk to each other, you will not understand this, then you will not understand the entire chapter. Okay? Then sit and cry. And then the actin. Okay? I am not drawing the glob, but just one or two just to make you understand. Okay? Actin and myosin. Done? Clear? Yes or no? Now, listen carefully. To keep all of these people in place, to keep all these muscle bundles in place, we have a connective, okay, a collagenous sheath, okay, a sheath like this, which is covering, which is actually present in our body, students. Okay, different types of fascias are there, but here you are talking about just fascia. Okay, there is visceral, parietal, deep structured. Okay, many types are there. So to keep all these muscle bundles in place, you have fascia which is going around. Okay, which is going around. So this is a, so far clear? So far clear? Which is a collagenous sheath. Okay, collagenous sheath. Okay, now let's start labeling each portions. Ready? Okay, now we are going to learn about the layering. Okay, we are going to learn about the layering. Now understand, students, the entire muscle, okay, the entire muscle will be lined by one layer. Okay, will be lined by one, okay, just like how it will be, no, like a plastic cover types, no, like that is going to look, but it's not like that. See, can you see one transparent layers? Can you see the one that he opened? Okay, so like that transparent layers are going to be there. Okay, so they're pulling out, you can see how the layering is happening. So same way here also understand that this entire muscle is going to be lined by what? I taught you people last time also, tell me. Like this, let's say I'm just drawing this dotted lines. Entire muscle is going to be lined by epiphyseum. Epimysium. Okay, epimysium. Correct? Agreed? Anyone who does not agree? If you don't agree, you should tell me the answer also. Okay, entire muscle, okay, this entire muscle is going to be covered by your epimysium. Okay, epimysium. Done? Okay, now going further, okay, this muscle bundles also will have a lining. This muscle bundles also will be having a lining, and that lining, okay, that lining is called, to not confuse you, I am just connecting these, removing this fascia from just this muscle bundle, okay, that you don't get confused. The entire muscle bundle has a covering called as the, okay, called as the perimysium, okay, perimysium. This is the most important part of this chapter. Very important part. Okay, perimysium. Very beautiful spelling all of you have put. Okay. Very beautiful spelling. I'm impressed by all of your spelling. Okay, perimysium. Now, when I go further, this muscle fiber also is covered. Okay, muscle fiber also has a covering. Okay, muscle fiber also has a coating or a covering, we tell, no? This one, like this. Okay, which is called the endomysium. Endomysium. So I've put all the three layers in yellow color so that you can clearly see it. Okay, you can clearly see it. So let's do a quick recap of what we just studied. Okay, this is a bone. To the bone, the muscle is attached with the help of a tendon. The entire muscle has a protective covering which is called the epimysium. Now, the entire muscle, if I see, inside the muscle, there are going to be muscle bundles which are irregularly shaped. Okay, muscle bundles are going to be there. Those muscle bundles are going to be called as fascicles. Okay, fasciculi, we can tell fascicles. Okay, now the entire muscle bundle is covered by a protective covering called as perimysium. Okay, perimysium. Then if I see, okay, if I see the muscle bundle, if I go into the muscle bundle, I will notice the muscle bundle has muscle cells or muscle fibers. Okay, what is it called? Muscle cells or muscle fibers. This is your muscle cell or muscle fibers. Okay, I should have actually drawn it here. I want to do more labelings. 1 second, students. I'll just remove endomysium. Okay, because I want to do the labeling there. Yes. Okay, 1 second. Give me just 1 minute. We'll just do this labeling later. Okay, so as I'm telling, you write it down. So inside the muscle bundles, there are muscle cells, or we can also call it as muscle fibers. Why can I call it fibers? Because they are long and slender. Okay, muscle fibers. Now inside the muscle fibers, there are more, okay, thin structure which is called the myofibrils. Okay, myofibrils. And if you go further, you will see your myofilament. Okay, myofilament. Now here you will see the thin and the thick filament, that is actin and the myosin. This is the best diagram that is possibly, you know, which can show you that, okay, this is the greater picture. Okay, this is the greater picture. Now clear? Muscle fiber is there, myofibril is there, then your actin and myosin is there, or you can write the myofilament itself. Okay, now to keep all this muscle bundle together, you have a collagenous sheath. Okay, a collagenous sheath which is called as the fascia. Okay, I'm just darkening it here. Clear or not? Put thumbs up in the chat box if all of you understood, or put done if you understood. Come on. Okay, there is fascia that is going to cover the entire layer. Correct? I think. Yes, those are collagenous tissue which are going to make sure that all the bundles are going to be in place and attached. Clear? Okay, so I'm going to label that as well. This is called your fascia. Done? Okay, now the entire muscle, your muscle cells, we tell, no, that is also covered by a protective covering. Okay, that is also covered by a protective covering which is called as what, students? Which is called as what? The endomysium. The endomysium. So covering that also, there is a layer called the endomysium. That's it. This is your entire thing. This is your entire thing. Very good. Done. Okay. Apsara, 3 hours maybe. No, Cappy, you continue from here. We're entering the main portions. Okay. Yes. Students, this is clear. Any doubts so far on this? Any doubts? No, right? Okay. Let's see here. Okay. Let's look at this. Can you see this? It is very fast, but you try to see it again and again. Okay, muscle, your entire muscle bundle, myofibril, and inside you will be able to see your actin and myosin, and these are your sarcomeres, which I'm going to teach you. Okay, these are your sarcomeres. I hope this works. Yes, look at this. Now, this is what we are going to learn. How the contraction is going to happen. No, this is what we are going to learn. Okay. Yes, perfect. Now, moving forward. Yeah, break will be there. This is done. Fine. Now, look at your Diagram given in your NCERT. Okay, diagram given in your NCERT. This is your entire muscle. These are your muscle bundles. Okay, can you see fascicles or muscle bundles is given, and can you see from the muscle bundle one muscle fiber is pulled out? See, muscle fiber or muscle cell is given. Again, it is called fiber because they are long and slender. Okay, long and slender. Okay, long and slender. Then students, sarcolemma and all of that I will teach you. Okay, sarcolemma, all of that I'll teach you. Okay, clearly you right now understand this much. Okay, now what is this sarcolemma? Okay, sarcoplasmic. Remember that whatever is related to muscle, we will use the word sarco. Okay, so the endoplasma, the endoplasmic reticulum, okay, ER, in muscles will be called as sarcoplasmic reticulum. Correct? Sarcoplasmic reticulum. The plasma membrane, okay, the PM I'm writing. Oh, no need, you people will think something else. Okay, you'll think Prime Minister. Plasma membrane. Plasma membrane of muscle will be called as sarcolemma. Sarcolemma. Important. Okay, important. Very nice. We reached the target. Good job. Okay. Next one. Endoplasmic reticulum clear. Plasma membrane clear. Cytoplasm. Okay. Cytoplasm will be called as sarcoplasm. Sarcoplasm. Okay. So here you can see sarcolemma they have told. What is sarcolemma? The plasma membrane. Okay, what is sarcolemma? The plasma membrane. Now, one thing you remember right now, okay, which will be useful for us in the later, is that this sarcoplasmic reticulum is the storehouse, okay, storehouse of calcium ions. Calcium ions. Students, after this class, you will just have to read NCERT once and you will be sorted, okay. You will be sorted. You will not have to study it over and over again because the chapter is very easy. Upon that, I will draw so many diagrams that make you understand, okay. Yes. Tongue twisting is happening. Okay. Now, most important. Now, students, now you understood that there is muscle. You understood there are myofibrils. You understood that these myofilaments are the reason for this light and dark striations. Now, who is the exact reason for contraction of the muscle? Who is actually the functional part of the muscle? That is going to be your sarcomere. Sarcomere is present between two Z lines. Okay, two consecutive Z lines, your sarcomere is going to be present. Now, I showed you sarcomere here. Look at this. So, if you see your myofibrils, you will be able to notice that there's one structure coming out and this is exactly what a sarcomere will look like, and they are the sole reason for your contraction. Okay, they are the reason for your contraction. So, now you people understood that your skeletal muscle, okay, or what we were talking about, these people have dark and light bands, okay. So, these people show striations. Yes or no? These people show striations because of mainly two things. A band, which is called anisotropic band. Anisotropic band, and the presence of I band, which is called as isotropic band. Okay, isotropic band. Now we are going to draw a sarcomere for you to understand all of this. Okay, someday I'll come. Okay, yes, I will definitely come. Thank you, Tanuja. Thank you, Madhumati. Okay, yes. Now look here. If you people scored really well, I'll be able to come to all the centers. Okay, I can tell that my students scored well. I need to go to all the centers. Okay, chalo, let's do this. Okay, now students, I am drawing one line like this. Okay, one line like this. Are they equal? Definitely not. 1 second. Yeah. Since I can't see if they are equal, I'm assuming they are equal. Okay, like the position I'm telling. Okay, they look almost equal. Okay, now inside this, let's draw a Z line. Okay, I'm drawing it little different this time. Okay, one zigzag line I'm drawing. This is the zigzag line. Okay, one more zigzag line. Okay, one more zigzag line. This is 2 zigzag lines. Yes, ma'am, 2 zigzag lines. Now in the middle, okay, in the middle, we are going to draw one thick portion. Okay, this is the thick portion that you're seeing here. Okay. Then on these, we are going to draw some thin lines like this. Okay, one here, one here. Same thing goes here. And one goes here. Okay, this is how it is going to look like. Okay, something like this. Okay, we're just drawing to make our work more easier and easy to understand. Okay, now let me just fill in these gaps. Okay, so it will be better. Now students, what have I drawn? I've drawn actin and myosin, which are your contractile protein. Okay, we will learn more about it. Don't worry, we are going to study the structure and everything. But first you should study the sarcomere. Okay, now same thing, we will fill in the myosin as well. Okay, myosin as well. Yes, this is how your myosin is looking like. Okay, done? Clear? Yes, done. Now, very importantly, now students, remember, I told you sarcomere is the unit that is going to help with the contraction. Now, exactly how will the contraction happens? The signal has come. Now, which is the line that will be ready to come forward? But before that, you should know, these are your 2 actins. Okay, these are your 2 actins. Okay, yes or no? Yes. Actually, the zigzag lines are too much down, students. Just give me 1-2 minutes. I'll just redraw this. Okay, I'll just redraw this. Okay, let's first draw actin and myosin. That is going to be easier for us. Okay, all this takes time to draw. That's why I'm telling you people don't have to draw, just try to understand. Okay, try to understand, but you have to give each other time to draw. Yes. Okay, now this is actin. Now remember that the actin, the actin will be kept in place. The actin will be kept in place with the help of a zigzag line. Okay, with the help of a zigzag line, which is called as your Z line. Okay, like this. One zigzag line will be passing, which is the reason for why your actin will be in place. Okay, so like this. No obedience, only the zigzag line has, just like you people. Stay here. Okay, this is how your zigzag line is. This zigzag line will make sure to keep this actin in place. Okay, this actin in place. Done? This is your 2 things. Clear? Now, in the middle, who is going to come? In the middle, your myosin is going to come. Yes, ma'am. In the middle, myosin is going to be placed. This is your big myosin, because myosin is the thick filament. Okay, myosin is the thick filament. Done? Diagram is almost done. Now let's go back to understanding each of it. Yes, so now you understood that these 2 are the actins, and the middle one is a myosin. Yes or no? Middle one is a myosin. So now this I'm labeling it as just A. Okay, this is also A, and this I'm going to label it as the Z line, because this Z line has so much importance in the contraction. Students, what have I drawn right now is a sarcomere. Now exactly understand, sarcomere starts from this Z line to this Z line. That means one sarcomere starts from one consecutive Z line to the next consecutive Z line. Okay, yes or no? Yes or no? What is wrong, Apsara? Nothing is wrong. Okay, so from one Z line to another Z line, this is called as a sarcomere, which is going to help with the contraction. Perfect. Now, to keep this myosin in place, okay, to keep this myosin in place, there is going to be one line which is called the M line. To keep myosin in place, there is going to be one line that is going to be called the M line. Correct? Yes, ma'am, M line. So we will label this also M line. Okay, now comes the important part. How is this dark and light band exactly formed? It is because of I band and A band, which I have written there. The full form I have written there. Anisotropic band and isotropic band. Now students, remember the area where only your actin is present. Okay, this portion is going to form the I band. Isotope, isotropic. What does that mean? Same. Okay, so this portion, okay, this portion where only actin is present, this is going to be where the I band is going to be present. Okay, one I band here, obviously another I band is going to be here. Okay, I band. Correct? I band and I band. Yes, sarcomere is the structural and functional unit of the skeletal muscle. Okay? Okay, got it. Now, here, this portion, okay, this portion where your myosin and actin is going to be there. So let's consider this, okay, where watch is there, no? This as the myosin, this as the actin. This portion will be having your A band. Okay, this portion will be having your A band. This is your A band. Clear? A band is called anisotropic. That means it is not just one thing, two different things are there, okay? Anisotropic. So I band and A band is the sole reason for why the bands are thick and thin. Now students, remember in the isotropic band you only have the actin. Now remember actin, thin filament. Thin filament. Correct? Who's going to be the thick filament? Obviously ma'am, myosin is going to be the thick filament. Okay, myosin is going to be the thick filament. Correct? Now we are going to learn some more regions. Okay, some more regions. Now students, the portion, okay, the portion where your actin and myosin is overlapping. Can you see actin is on the top, myosin is down. This portion can be called as the O zone. This portion can be called as the O zone. What zone? O zone or O band also you can tell. Okay, O zone or O band. Here also you can see O band or O zone. I have just put O there. Can you see? Yes? Very good. Okay. Which place? The portion where only your myosin is going to be present. Okay, the portion where only your myosin is going to be present, that is called as your H zone or Henson zone. H zone or Henson zone. The portion where only your myosin is going to be present, that is called as H zone or Henson zone. Clear? Yes or no? Okay, perfectly clear. Who is telling no ma'am, yes ma'am? Who are the ones who are crying like that? H zone or Henson's zone. Okay, this whole thing is the A band, student. Okay, this whole thing will be the A band. Okay, from here till here almost will be your A band. If I draw too many lines, no, the problem is it will become very confusing. That is why I avoid drawing that many lines. But understand, these two white lines, okay, indicate your entire A band. Correct? The portion where only myosin is going to be present, that is going to be your H zone. The portion where actin and myosin is overlapping, it is going to be called the O band or the O zone. Okay, O zone only we'll tell, overlapping zone. A band you understood, from this white line to another white line. Okay, this white line to another white line. Done? Any doubts? Who is the one who is telling no, yes? I hope it's clear. Okay. Now, ma'am, why did you teach all this? Okay, why did you teach about the H, I, A band, all these bands? Students, understand, when contraction, when your nervous system or when your brain is signaling that please contract, who is the first person who is going to move forward? Who is the first person who is going to move forward? It is going to be your Z line which is going to move forward. And when this Z line move forward, you should know which are the things that are going to move, which are the things that is going to be constant. Okay, which are the things that is going to move, which are the things that are going to be constant. So looking at the diagram, all of you try to tell me which are the things that are going to move. So I'm going to write it here in the corner. Okay, I'm going to write it here in the corner. I'm telling that when contraction happens, okay, when contraction happens, which is the first thing that is going to move? All of you tell me in the chat. Ma'am, Z line is going to move inwards, right? It is going to move inwards. I cannot write the whole thing. We will write it in the next page. Okay, now I just want you people to see the diagram and tell me the answer. Very good, very good. Apsara, no, very good. Pallavi, Roshni, Kharti, Z line. Okay, Z line, very good. Now, the next thing, when the Z line is going to move forward, is A band going to be constant or what? Okay, here understand that here A band is going to be constant. A band is going to be constant. A band is like, I am not going to move. If you want, you move. I am not going to move. Okay. Wow. Okay, how nicely people are welcoming others. This is why I love my GMAs. They're the best students ever. Okay. Yes. So, did he tell GMA zoo? Okay, I take everything what I just told. Okay, no best, nothing. I take everything. Okay, back. Okay, A band is constant. Students, with A band, M line also will be constant. M line also will be constant. Okay. Whole attitude they have. They are like, no, I will not move. Okay, if Z line has come forward, let him deal with it. I am not going to deal with it. Okay, I'm not going to deal with it. So Z line will move inwards. A band is going to be constant. M line is going to be constant. Now, think carefully and tell me, will ozone increase or decrease? Next question to all of you. Ozone will increase or decrease? Very good. Very good. Hensen zone is going to reduce, okay, drastically, right? Because this is going to come more and more. So here, the myosin area is going to be less. So Hensen zones will reduce. So H zone also will reduce. H zone reduces. Then what, students? Ozone will increase. This is the only person who is going to increase. Okay, ozone is the only person who is going to increase. Very good, Pallavi. Very good, Madhumita. Okay, very good, Suvra, Teju, Arya, the person who told zoo. All of you, very good. Okay, yes, because overlapping zone is going to increase, because these two, let's say from both the sides someone is pushing my elbow. Overlapping zone is going to increase. No, overlapping zone is going to increase. Okay, overlapping zone is going to increase. So ozone will increase, H zone will decrease. Obviously I band also is going to decrease. So this is what happens. Okay, during your contraction, so many things are happening. Yes, H band almost disappears, or we can write reduced only. Okay, almost decreases. O band is the only one that decreases. I band also decreases, students. Okay, ozone is the only one who increases. Obviously I also will decrease. I band decreases. I'll just put the arrow marks. Clear? This is what happens when contraction happens. So sarcomere is the reason for the contraction, and in the sarcomere, which are the lines that decreases, increases, you have to know, because questions keep coming from this portion. Okay, question keep coming from this portion. Is this clear to all of you? Yes or no? Is this clear? Let me see what is the next one. Yeah, this is the one. Okay, this is what we drew, students. Myosin, and this is your actin, and this is the one portion of the Z line. Can you see? See contraction, how it is happening? Can you see how the Z line is moving forward? Yes, is it visible? Yeah, so look here. This is your zigzag lines, no? So one more will come. See, now the Z line is moving forward. Can you see so many of the zones are decreasing? Okay, so many of the zones are decreasing. Okay, done. All of you done, or only few? Okay, now actually, okay, I'll just duplicate this and I will write all the names of the bands. Okay, yes, otherwise what happens, student who is seeing the image will not know the names. Okay, yes, we'll just write the full form, students. Many of them will be there who does not know the full form, so we will just write that. Okay, so yes, so we will start with your Z line. Z line, you know, it's called holded by the actin and all of that. M line and all that. Okay, I band, we will start with I band. Okay, isotropic. Okay, isotropic. A band and isotropic and isotropic. Then what do we have? H zone Hensen zone. Okay. O, overlapping. Overlapping. O we did, H we did, M line, A band, everything we did, no? I, Z line. Okay. Yes. Hensen's. Okay. Thin, thick bands. Yes, very good. Thin, thick bands. Okay. Now, students, understand this one thing. Okay. Understand this one thing. I think Z line, H line. Okay. Okay. So, in one sarcomere, they can ask you a question. This one sarcomere, okay, what is the entire, okay, if I have to tell you what is one sarcomere has how many I bands and A bands, your answer should be one sarcomere, okay, one sarcomere has how many, students? Look here and tell me. Always remember, your sarcomere is from one Z line to a next Z line. This is one Z line, this is one Z line. So I cannot count what is this side. So I am going to tell you this is one I band means what? This whole thing is an I band. So I can only take half of the I band. Yes, ma'am, you can only take half of the I band. Here also, ma'am, you can only take half of the I band because this entire thing is the I band. So one sarcomere is half I band plus half I band plus one full A band. Yes, ma'am, one A band. So in total, okay, this half and half you should know. Don't be like, ma'am, why you writing half plus half? Why don't you write one full? You should remember that. You should know that why I have written one half half. Okay, half half. So that totals to one I band plus one A band. Clear? So one sarcomere is equal to one I band plus one A band. Done, done, done. That is your, okay, that is the simplest thing that you have to remember. Simple thing that you have to remember. Yeah? Okay. Who's asking something? Okay. Okay, done. Yes, two halves and one full A. Very good. Okay, now structure of myosin. So now we are going to learn about the contractile protein. So these are the proteins, right? Now we just learned about actin and myosin. That, ma'am, actin is there, which is, actin will be kept in place with the help of Z line and myosin will be kept in the place with M line. But then we do not know how this contractile protein looks like. What is the exact reason for their contraction? Which structure are they binding with? All this also you have to know. So remember that your myosin is made up of a monomer called as meromyosin. Okay, meromyosin. So when we learn about the structure, remember about meromyosin. Monomer and all, I don't have to teach you people, no, chemistry and all you studied. So monomer of myosin is your meromyosin. Now students, remember this structure is made up of 6, okay, it is made up of 6 polypeptide chain. 6 polypeptide chain. And out of this 6 polypeptide chain, you will have 2 long chains and 4 short chains. 4 short chains. Yes? Now, this 2 long chain and 2 short chain is very important for you to know because that is what is there in the structure. So students, we just draw like this, like this, like this, and like this. We draw one more like this, which is almost similar, not very similar also, but yeah, like how we draw our DNA double helical structure, no, like this. Okay, this is how your myosin is going to look like. Okay, very diagrammatic representation of it. Okay, so here this portion which you're seeing is the head region. Okay, this is the head region, and this head region has one of the most important things that you have to remember for the contraction or your sliding filament theory, right? So this portion, this is called as your tail. Okay, this portion, this entire portion is called as the tail. This portion, right? Tail. Now, one question has been asked, every single or mostly repeated from this question is that, is this tail, okay, light meromyosin? So remember, depending on the weight, molecular weight, they have been divided into light meromyosin and heavy meromyosin. It's obvious, no, your head is going to be heavier, right? So here also remember like this, your tail is going to be made up of LMM, that means light meromyosin, light meromyosin chain. Got it? Light meromyosin. Light meromyosin. The tail is made up of light meromyosin. Very good, the ones who are answering. Next, the head portion, students, very importantly, the head portion has two regions. Okay, the head portion has two regions. What are they? It will have, obviously, if myosin is there, it has to attract or it has to bind with whom? To actin, because these two are the contractile protein, then only contraction will happen. So on the myosin head, there is going to be ATP binding site, I'll tell you. Okay, there is going to be an actin binding site. Okay, there is going to be an actin binding site, right, where the actin will come and bind, obviously. Okay, actin binding site. And on top of the actin binding site, there is going to be one ATP binding site, okay, or a ATPase site. And you know what is the purpose of it, right? Understood? Yes, ATP binding site. Very good, very good. ATP binding site. Let's label it. So this is your actin binding site, which is very important. Okay, and this is your ATP binding site. Now students, remember, there is a short arm. This portion is the short arm plus the head. Okay, so from here up to here, we will have the heavy meromyosin. Heavy meromyosin. Got it? That is going to be head plus the short arm, or we can tell the cross arm. Okay, so this portion, this is definitely your head, you know that. Okay, so that is two portions, head plus the short arm. Together will be the heavy meromyosin chain. Any doubts in the structure? Please ask me right now. Heavy meromyosin is clear. So tail is like a fibrous tail and head is a globular head. Okay, head is a globular head. So I'm just going to write it here. You have the tail like a fibrous tail and you have the head, which is a globular head. And this head is very important for our contraction to happen, because on the globular head you have two things which is very, very important. One is an actin binding site. One is an actin binding site. And one is an ATP binding site. And this fellow is the villain in the story. Okay, this fellow is the villain in the story. Yes or no? Yes, ma'am. Done? Those who are attending for the first time, you definitely can put the comment, okay, how the class was and what is it, right? What is it? Okay. Yes. Now, next is the structure of actin. Now, students, structure of actin, what exactly here happened is, it was globular actins. That globular actins later got vital polymerization. No, it has happened to form the fibrous actin. Okay, so we will start with the globular actin and then we will go to the fibrous actin. So globular actin means what? Just some circular structures. Okay, some globular structures. So this was your first globular actins. Okay. Globular actin. So this also question has come. G-actin and F-actin question has come. I have added that question today also. You can see the question once I finish the topic. Okay, once I finish the topic, you can definitely see the question. Okay, so globular actins or G-actin we were telling. Globular actins or the G-actins. Okay, later they will undergo polymerization and they will become what, students? They will become the fibrous actin. Okay, they will become the fibrous actin. Okay, yes. Now, remember, when I am drawing the fibrous actin, I am basically going to draw something like your infinity sign. Okay, because similarly the structures, I wouldn't say it is exactly like that, but something similar to this. Okay, now this structure is very important because you should remember that this structure is going to have these globular actins only arrange themselves in a fibrous form. So we can call this now as F-actin. Okay, we can call this now as F-actin. I will show you the structure given in your NCERT also, because that diagram they can ask you to label the portion, and actin question keeps coming in all the mock papers. If my students are part of DNA or have written mock paper, you will know that this question is one of favorite question. Okay, everyone keeps asking this question. Yes, so students, see, now I've just drawn circular structures and I've almost done your actin diagram. Okay, yes. So now this structure, more than the structure, what lies inside the structure is very, very important. Okay, what lies in the structure is very important. So I will call it the F-actin. Okay, I will call it the F-actin or the fibrous actin. Now, on this structure, there are two main regulatory proteins. Okay, what are they? Two main regulatory proteins. One of the regulatory protein will be present in proper intervals. Okay, proper intervals. That is going to be a 3 structures like this. Okay, 3 structures together like this is how they are going to be present. Is it visible, or should I draw it with some other color? Should I try with blue? Okay, they are going to be present in proper intervals. Okay, yes, someone is asking a question. Oh yeah, so sorry, I forgot about it. Okay, students, the 4 short chains, okay, 4 short chains are going to be present like this. Okay, okay, wait, I'll draw it with the, I almost forgot about it. Your 4 short chains are going to be present like this. Okay, they're going to be present like this. Those are your 4 short chains. Got it? Vaishu, thanks for asking, dear. I almost forgot about it. The 4 short chains are going to be present like this. Okay, that is your short chains. Now, moving forward, F-actin. Okay, F-actin, as I told you, there are going to be 2 regulatory proteins. In that, one of the regulatory protein is going to be placed at proper intervals, and they're going to be 3. Okay, 3 circles are going to be there, and what are those 3 people are also very, very important. Now, do you see that? Yes, ma'am, I do see that. In between, okay, in between lies one thread-like structure. Okay, thread-like structure like this, and this is also one of the regulatory proteins. Now, why am I calling them regulatory proteins? Because they are going to control if contraction has to happen or not. Okay, they are going to be the controlling factors about the contraction. Now, students, let's just draw this. Okay, this fellow separately. This fellow separately. Right? This person has to be drawn separately for you to understand what these 3 structures are going to look like. Yes. So, students, remember that your 3 structures, you have one on the top, you have one here, okay, like this. Like this. Like this, 3 structures are there. Visible? 3 structures are visible? Yes? All of you tell me in the chats what's the answer also. Most of you know the answer. Most of you know, I know all of you know the answer. Obviously, one time at least this chapter revision would be done. Yes. Tell me the answer for the students. What do we have in this? TPC. Yes. Okay. Tell me. TPC, TPI, TPT. Right? Yes. So here what you have to remember, the most important, oh no. Okay, most important is your TPC. TPC TPI and TPT. Okay. Now students, remember this TPC is the most important for you. Why ma'am TPC is so important for me? Because this is the place where your calcium is going to come and bind and also allow for contraction to happen, which you will understand in the next. So this chapter goes in such a way that, you know, so many things are mixed, but obviously we have to teach it this way. We cannot teach this first and then the sarcomere or the other things. So students, this is your calcium binding site. Okay. This is your calcium binding site. This is your troponin. Troponin. Okay. Troponin, and this is your actin inhibitor site. Okay. Actin inhibitor site. Now, remember that I told you there are two regulatory proteins. Ma'am, then what about the other one? Right? What about the other one? So, remember your two regulatory proteins, okay? Two regulatory proteins are going to be called regulatory, okay? Are going to be called, one is called troponin, and one is tropomyosin. Tropomyosin. Clear? All these notes will be available on Vedantu NEET English Telegram as well as Biology by Gopika. So make a note of this. Okay? Yes. TPI, TPC and TPT. Now is it clear? Niti, it is clear? See, TPC is your calcium site, TPT is your troponin site, and TPI is your actin inhibitor site. And they are going to be present at every single interval, and they are going to have also tropomyosin. This line-like structure is going to be tropomyosin. So students, these two people, that is your both the regulatory proteins, are going to be the main reason for controlling the contraction. On the actin also there is a myosin binding site. Remember this. Okay, on the actin also there is a myosin binding site. Okay, actin also has a myosin binding site which is masked. Okay, binding site that is masked by regulatory proteins. You cannot see it, that's why we do not label it also. But remember, on actin also there is a myosin binding site which is masked by the regulatory protein. Yes. TPC da, not T and T. Write off ENT only then last. ENT, gynecologist, everyone you write. Okay, clear? Fibrous, everything is clear. Any doubts so far? If you have doubts, you should ask now, not later. Perfect. Okay, good job. Students, that's short arm. You can also call it as cross arm. Okay, so don't worry about it. Yes. So these are helically coiled structures, but before they were globular, and then remember polymerization happens and they become like this. Okay. So polymerization happens. Okay, and they become F-actin, which is more helically coiled structure. Okay. Helically coiled. This is your whole story. Now with this story only you will be able to understand how contraction and relaxation happens. Okay, how contraction and relaxation happens. Let's see, ready all of you? Yes, full energy is there. Let's put some fire emojis in the chats. Come on. Let's increase the energy in the chat box. For calcium binding. Okay, for calcium binding, because this is the place where the calcium from the endoplasmic reticulum will come and bind. Don't worry, you will understand about this TPC, TPT. Okay, all that. In that you just remember only TPC. Just remember that the troponin has a calcium binding site, and that is the reason for unmasking of the myosin binding site. Okay, yes, there is actually more in depth to study about this topics, but right now we are not studying that much. Okay, we are not studying that much because obviously you people don't have time, and if we read something out of NCERT, you people will be the first one to cry also. Okay, first one to cry also. Done? Made a note of everything? If yes, we will move forward. Yes. Done. Okay. Now, this is the structure given in your NCERT. Okay, structure you can see, beautifully, clearly you can see. So this is your actin. Can you see how the actin is? It is like double helical structure, in between the myosin and the troponin is running. So students, this is the most asked question. This is a PYQ and this is seen in most of the mock papers. The question will be, they will ask identify A, B and C. Okay, any order they can give you, A, B, C. And you will have to identify it. Now a student, remember the image will not be this beautifully given to you. It could be a black and white image, it could be a color image, but mostly it will be a black and white image. So be very careful when you're labeling F-actin and troponin. Many students do not understand this. See, this is your F-actin because this is entirely in a fibrous situation. Most of the students will go and tell globular actin. Globular actin is before polymerization, when they are all scattered. After polymerization, we call them as fibrous actin. Okay, so you're going to label here F-actin, not G-actin, not G-actin. And here students, it is tropomyosin. The thread-like structure that you're seeing is the tropomyosin. And here you see the troponin. That's why I told you, you don't worry about TPT, TPC site. You just remember that troponin has a calcium site. Okay, calcium site. Okay, it's fine, right? Just blinked for a minute. Yes, many questions are there now. Yes, perfect. Okay, troponin and tropomyosin and troponin. Okay, now here students, actin binding site. Can you see actin binding site and ATP binding site? So you notice that your ATP binding site is there, actin binding site is there, but again this structure, this portion is the head, this is your cross arm or the short arm, and this is your tail, which is the light meromyosin. Okay, LMM. Depending on the molecular weight, this categorization is done. This question also has come a lot of time if these two are a part of HMM or LMM. This question also has come a lot of time. Again, a PYQ. So remember these two diagrams easily. 1 4-mark question can easily come. Okay. Oh, Bunny, enough. Friends, come on, let's make the like 150. Fast 150 likes. Fastest fun 150 likes. Okay, I need energy. For me to work more, I need energy, right? Yes, done? Okay, let's move forward. Okay, now comes the most important part, how contraction happens and how it exactly happens. First, I will teach you with the help of this diagram. Then I will draw a diagram and help you understand. Okay, now I told you students, your nervous system, your muscular system as well as your skeletal system have to be in coordination with each other for any kind of contraction or any kind of movement to happen. So now you will understand how it exactly happens. Look here, all of you. Look here, all of you. This is your nervous system bringing the impulse. Can you see? Motor neuron axis. Okay, listen to me carefully. Now, when this axon is bringing it, you will see the motor end plate. Okay, this axon is bringing the signal to the muscles. Okay, muscular, neuromuscular junction. That means where the neuron and the muscles will be attached, neuromuscular junction. Now, students, look at this diagram. Focus on this diagram. Can you see? One neuron, that is the axon ending, has come and attached to the muscle. This entire portion is a neuromuscular junction. What exactly is happening inside the muscle is what you have to notice. See, can you see acetylcholine, that is your neurotransmitter, got released. It is being accepted by the receptors which are present. After accepting the thing, this is going through a T tubule. Can you see a T-shaped tubule? With that tubule, the sodium influx is happening, because of which your calcium will be released from the endoplasmic reticulum, and that calcium will go and bind with your TPC site, and you will see that the myosin is ready to bind with the actin. Now, this is the diagram. Now, I'll teach you in my way. Okay, I'll teach you in my way. Okay, what is exactly happening? Okay, now let's say students. Okay, this is your muscle. Okay, this is your muscle. This is your muscle. Now, in the muscle, what is happening? I already told you these structures. Okay, this neuromuscular junction, this is from here. Who is coming? Axon is coming, and with the axon terminal, okay, it is going to attach here. Right, it is bringing the signal for us. Okay, it is bringing the signal for us. Correct? Now, in the axon terminal, what do we have? In this axon terminal, what do we have? Ma'am, we have our neurotransmitters. Okay, we have our neurotransmitters. So remember, let's say if I just draw this a little below, like this, okay, just a little below. Okay, this is the T tubule, students. I have to draw it a little above, so for you to show, I have to show you the receptors also, that's why. Okay, let's say there are receptors present like this. Why these receptors are present? So that they can take this acetylcholine and do an influx of sodium. Okay, some receptors I have drawn. Can you see the receptors? Yes, I can. Yes, ma'am, you can see, show the receptors. Now here, let's say from here, the axon is coming. Okay, at the end of the axon, there is going to be axon terminal, and inside the axon terminal, what is there, students? Acetylcholine is there, which is one of the most common neurotransmitter. Okay, one common neurotransmitter. So acetyl neurotransmitters are ready to get released, and these neurotransmitters are going to get released and they are going to be accepted by the receptors. Correct? They are going to be accepted by the receptors. This is clear? Clear? Yes. So here, this is your axons. Okay, this is axon. And this is your neurotransmitter. That means your entire, students, your synaptic bulb, we tell, no, synaptic bulb. So that one, okay, inside that only your neurotransmitters will be there. So that is going to, so here I will write AC. No. Okay, here I am writing ACH, that is your acetylcholine. Okay, which is a neurotransmitter. Okay, neurotransmitter. Now, where is all of this happening? This is happening in a neuromuscular junction. Okay, now students, as the receptor takes in the acetylcholine, what is going to happen? It is going to lead to influx of sodium. We have studied this in action potential also. We have studied this in neural control and coordination. It's easier to understand, no? That as they accept, there is going to be an influx of sodium ions inside through this T tubules inside the muscles. Okay, inside, right? So sodium influx, sodium influx, sodium influx. Okay, here also same thing is going to happen. Everywhere sodium influx is going to happen. I'm just drawing the arrow mark. You should understand that it is sodium influx. Now students, so much sodium influx, what will it do, ma'am? Okay, what will it do, ma'am? That is important, no? So much sodium influx will lead to what? For that, you have to understand that your endoplasmic reticulum, right? Your endoplasmic reticulum, which is your sarcoplasmic reticulum. Okay, they are present, right? They are present inside the cell. They are present inside the cell. Yes, ma'am, they are present inside the cell. Now, inside them, there is a storehouse. So, I'm just going to draw a circle oval structure, students. Otherwise, it will become too complicated for me to show the calcium thing. So, this is your sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. Why is this not moving? Can I move it? Yeah. Okay, like this sarcoplasmic reticulum is there, which I told you they are the storehouse. They are the store calcium. Now, because of so much influx of sodium, this calcium ions will be released from the sarcoplasmic reticulum. Clear or not? From the sarcoplasmic reticulum, your calcium ions will be released. Okay, will be released because of the continuous influx of sodium. Calcium ions will be released from the SR I am writing, that is sarcoplasmic reticulum. And now where are they going to go? They are going to go all the way to your TPC. Okay, this is your actin. Okay, on the actin, what is there? Your TPC, troponin is there. This calcium is going to come and bind with the TPC and do conformational change. What is it going to do? Conformational change. Correct? So this calcium will come and it will bind with the TPC. Okay, bind to the TPC. Basically, students, what is happening here? They are distracting. They are distracting, or they're trying to do a conformational change. Telling that, hey, you go and talk to him. Suddenly, I will try to bring myosin inside. So basically, what is happening? Actin, on the actin, there is myosin binding site, but it is not visible to us. It is masked. With whom? With the help of myosin, tropomyosin and troponin. So what we have to do, with the help of this calcium release, now it will bind to the TPC, because of which what will happen, students? This troponin and tropomyosin will move. They will get distracted. So here they will form a conformational, okay, they will create a conformational change. Conformational change. And this conformational change will lead to unmasking. Of myosin binding site. Myosin binding site. Is this clear to all of you? This is one of the most important topics, and it is not written very organized in NCERT. So that's why you have to study it like this. Okay, please tell me if you understood. Please tell me if you understood. Students, basically what is happening, let's learn it like a story. Okay, let's learn it like a story. So let's say, okay, let's say myosin and actin is not able to meet each other. Okay, let's say they are going to be, they're going to get married. Okay, they're going to get married. Okay, if they get married early, then everything is going to happen. That is, contraction should happen of the muscles. Okay, so this actin has two brothers, troponin and tropomyosin. Now, this person is not allowing actin to meet myosin. So, what we are going to do? We are going to make a change. How is that change going to happen? When an axon is bringing an neurotransmitter, and when the neurotransmitter, that means basically when stimulus is coming or a signal is coming, and that neurotransmitter will get released, and the receptors will start accepting this acetylcholine, and once they accept this acetylcholine, there is an influx of sodium into the T tubules which are present in the muscles. Okay, these are called as T tubules. T tubules. Okay, these are T tubules. These T tubules, there will be an influx of sodium. That influx of sodium will lead to the sarcoplasmic reticulum, because sarcoplasmic reticulum is the storehouse of calcium. It will allow sarcoplasmic reticulum to release the calcium ions, and this calcium ion will be like very chalak. He's like, I am your best friend. Whatever happens, I will make you meet actin. He will tell to myosin. Okay, this calcium will go and deviate or distract, we tell, no, like that. It will deviate your troponin and tropomyosin, and that will lead to a conformational change, allowing unmasking of myosin binding site. Okay, very sad story, but we have a happy ending to it. Okay, happy ending to it. It will deviate troponin and tropomyosin. Troponin and tropomyosin. Is this story clear? If yes, put done, done. Okay, put done, done in the chats. Fast. Done, done in the chat. Quickly, and let me see how many people have marked your attendance. Understood? Done in the chats. Yes, Joshi, Soni, Ratis, Karti, Suvra, Saravanan. Yes. Done. Apsara. Okay. So this is the story. Okay, this is the story of whom and whom. It is the story of how actin is going to be visible. Okay, that means myosin binding site is visible on actin. Okay, so this is the story of unmasking myosin binding site. Unmasking myosin binding site on actin. Clear? This will only allow them to bind, otherwise the binding will not happen, because here the masking has happened. Okay, binding will not happen. Now students, with this, okay, with this, if you understood, we will just see this also, okay, we will just see this image. So what is happening? Can you see this T tubules? How they will be? T tubules will be present, okay, in the neuromuscular junctions. So students, here you see one axon, okay, this is the axon. It has attached to the muscles, okay, that has led to the release of neurotransmitter acetylcholine. That is the second step. This acetylcholine has been released, and can you see receptors are accepting it? After the receptors are accepting it, you will see an influx of sodium in the T tubules. That will lead to the release of calcium ions. So this one, what you're seeing is the release of calcium ions from the sarcoplasmic reticulum towards the actin. This will go and bind with the troponins here, okay, they will go and bind with the troponin, that is TPC, and allow. Can you see myosin is already ready? Myosin is already ready by releasing ATP. He has already hydrolyzed ATP and he has made it to ADP and PI. Clear? Now, if we go here, okay, look at this, students. Can you see? This is your myosin. And what you're seeing here, this pink color structure is the head structure. And can you see how this Z line is holding the entire actin? So whatever I am teaching, this is how the representation is. Okay? Yes? Right? You can see, how it is happening. So you will notice that this pink lines, okay, this pink line that you're seeing is actually the myosin, and these are the heads which are stretching out, okay, standing out. And then you have this actin. Can you see? They're fully filamentous structure, and the Z line is going to move forward. How this happened? Only if the actin and the myosin binding site are visible to each other, Now look here. Now you look here. Can you see calcium ions? They came and bound with the troponin. Automatically myosin binding site got visible and myosin is ready to bind. All of you see this for 1 minute. See. What happened? Neuromuscular junctions, acetylcholine got released. They got accepted by the receptors. That has led them to know that, okay, it's time for releasing calcium. Okay, calcium has gone and bound with TPC, and you can see one yellow color myosin binding site visible. And automatically the myosin started attaching themselves. Okay, yes? Any doubts? With this, I think it should be super duper clear. You are the one who told zoo. So you are going to sit in one corner. Come on, students. Done? Yes, Pakka? No doubts, no? This is also clear? Yes? Okay. Can you see how it's happening? The tubules, can you see? Chalo, you people understood? Okay. Now we will go forward. Okay, now comes your sliding filament theory. Students, after sliding filament theory, I'll give you a break because after that we have to start skeletal muscles, right? So sliding filament theory, look here. Now, what did we do? We did unmasking of myosin binding site on actin. Now we have to unmask whom? In the chats, all of you tell me. Whom should we unmask? We have to unmask on the myosin, we have actin binding site. Two people have to meet, no? So two villains we removed, we got deviated, we did conformational changes and we moved that person. Now we have one villain here, that is your ATP binding site. ATP is sitting on myosin head and telling that I will not move. For that person to move, you have to do hydrolysis of ATP. That means you have to break down ATP to ADP and PI. Okay, so look here, the first process that is going to happen in this whole, thing is going to be, students, where you hydrolyze ATP to ADP and PI. Okay, normal thing, no? Normal thing. ATP to ADP and PI. Okay, full tongue twisting is happening today. Okay, PI. Now what is going to happen, students? Obviously, because of this, definitely myosin, on the myosin, the actin binding site was visible because ATP went. ATP got broken down. That led to cross bridge formation. Okay, this is like two countries. Okay, cross breed formation, like that only, cross bridge. Okay, because on the actin, myosin binding site is visible. Now the job is to make sure that myosin will be able to bind. For that, what we had to do, we had to remove this ATP, or we had to hydrolyze this ATP. That led to the cross bridge formation. Now when the cross bridge formation happened, okay, myosin and actin bound. Everything is going nice, okay, actin and myosin bound. But this ADP and PI, which is there on the myosin head, They are like, why should I be here? He already hydrolyzed me. I don't want to be here. So he will, that ADP and PI, he, she, I'm telling just to make you understand, okay, like a story. ADP and PI will remove, okay, will move out of the head, or we call that as power stroke. We call that as power stroke, where you will see that the ADP and PI has come out of the head. If something comes out of your head, okay, like your brain, if it comes out, okay, what will happen? You will move like this. Same thing happened here, students. Let's say my hand is there. This is the actin and this is the myosin. They bound. This is cross bridge formation. Now my myosin, okay, this is the myosin head. From here, ADP and PI moved. Because of this, what happened? Can you see a space between both of us? I look like one, one person who is like trying to do some aerobics or, some weird yoga asanas in the class, okay? It's like this to make you understand what and all I'm doing, you see. Okay, so again, actin, okay, myosin head. On the myosin head, you have ADP plus PI. Okay, now cross bridge formation has happened. You will not see any space between them. But after some time, ADP and PI has to be removed. So ADP and PI will be moved. Okay, removing time, a power stroke will happen, because of which myosin head will move from actin. The moment myosin head moves from actin, or actin slides over myosin, thin will slide over thick or thick will slide over thin. Obviously, thin will slide over thick, no? So, because of which the thin filament, that is actin, will slide over the myosin, and because of that gap, next ATP will come and bind. Is this clear to all of you? So, because of the power stroke, ADP and PI moved, and that moving led to a space between them, and immediately next ATP came and bound. Done, done, done. Is this clear? Is this clear? It's a very simple concept, but people complicate it, but you don't have to complicate it this much, okay? So, here also you can see, students, I just put the number, okay, number. Okay, yes. So, what is going to happen, students? Myosin. So, myosin and actin will bind together. How this happened? Because of ATP hydrolysis. After that, what has happened? Power stroke has happened. See, that means cross bridge formation. After that, power stroke happened. Myosin's head moved this side. ADP and PI rushed outside. After the power stroke, what happened? There was a space created between them. Immediately, an ATP came and bound. Okay, an ATP came and bound. Again, the process is going to continue, and that is how the sliding filament theory is going to continue. Any doubts in this? Any doubts? No, right? So, first what is happening, students? The first step is going to be, we have to hydrolyze. Okay, hydrolysis of ATP. That means ATP will become ADP plus PI. Okay, that means ATP has gone from myosin head. Problem is almost solved. Okay, almost, we can tell, we cannot tell full solved. Okay, almost solved. Now, what is going to be the second step? Ma'am, second step is going to be that now that ATP went, this is going to lead to cross bridge formation. Between whom? Cross bridge between myosin and actin. Okay, myosin and actin. Yes or no? Yes, ma'am. Myosin and actin will form cross bridge because they are very happy. Right. Now, what will happen? After that process, just write the cycle and keep next to the diagram itself so that you do not have to get confused. Okay? Okay. Now, so cross bridge between myosin and actin has happened. After that, what will happen, students? Power stroke will happen. What is power stroke? Where the ADP and PI. Okay? So, from the myosin head, the ADP plus PI will move out. Okay, will move out from myosin head. This is leading to power stroke. Okay, power stroke. Now, because of the power stroke, what has happened? Definitely the power stroke created a space. Okay, created a space. So, because of this, new ATP will bind to myosin head. So, this will also lead for actin to slide. Okay, actin to slide. So, here it will lead to actin to slide over the myosin, and this is exactly called as your sliding filament theory. Understood why is it called sliding filament? Because actin is sliding over the myosin, and that is the reason, and it is exact timing that when contraction and relaxation should happen. Okay, contraction and relaxation should happen. So, here also If you go to the previous one and see, you will be able to understand. Okay. Can you see how the sliding is happening? See, ATP, new ATP will come and attach, okay, as the power stroke happens. It will be hydrolyzed, ADP plus PI, it will attach with actin, it will slide, move, next ATP will attach. Just look at this for 2 minutes, you will understand. Yes, movement, again, new ATP attached, actin will move forward. Again, hydrolyzed, attached, power stroke, moved. Clear? This is what is happening. Okay, this is what is happening. Okay, done. Okay, this is your sliding filament theory. So always remember, students, thin filament will always slide over the thick filament. This question was asked and many students made mistake. I was really shocked. Many students went wrong in this. So remember, thin filament means always remember actin, thin filament. Thin filaments slides over thick filament. Thick filament. Okay, thin filament slides over the thick filament. Yes, so this is your sliding filament theory. So now students, remember, we will just so we have understood what is locomotion and movement, right? We understood what is locomotion and movement. After that, what did we understand? I don't know if it will work. Okay, this will not work. Okay, then we understood the different types of movement: amoeboid movement, ciliary movement, muscular movement. Under amoeboid movement, you have to remember WBC and macrophages. In ciliary movement, you have to remember lining of the fallopian tube and lining of the bronchi and trachea. Muscular movement, you have to remember your limbs, jaws, tongue, 639 muscles are there, and all of them are going to help us in the movement. Okay, flagellated movement, you have only one example, that is going to be the sperm. Now the properties of muscle, the most important property, or the 4 of them, are EEEC: excitability, elasticity, extensibility and contractibility, and we know the answer for each of the terminologies, also the meaning of it. And also remember that your nervous system, skeletal system and muscular system has to work together for any coordinated movement to happen. Okay, then moving forward, we learned the different types of muscles: smooth muscle, cardiac muscle and skeletal muscle. With the diagram, we understood what is the most important features. We study all this in structural organization, so this is just like an overview. Okay, then we understood what is the skeletal muscle and how is the diagram of it and what is all present inside this. So this was the foundation for us, right? So we understood about epimysium, perimysium and endomysium. We understood about fascia, fascicles, muscle bundles, actin, myosin, everything we understood in this diagram. Then when we move forward, again NCERT diagram, then we understood what is the sarcomere and which are the things that are present and which are the things that are not present, and also which are the ones that are contracting, decreasing in size, increasing in size. When a contraction happens, which are the things that are constant. Everything we have written it here in one small box. Okay, and we also wrote that what are the full forms of each of it. And students, remember, in PYQs the question has been asked about contracted, relaxed and maximally contracted. All if you remember that diagram, remember contraction. So when your sarcomere is contracting, I can't draw that. Okay, you will see the normal sarcomere, then you will see the contracted sarcomere, then you will see maximally contracted sarcomere. It's in your NCERT. That diagram they have asked as a PYQ, so look into that diagram as well. Then we understood the myosin, diagram, that is tail, head, everything, and 2 long chains, 4 short chains. These white ones are the short chains, these are the long chains. Okay, and we understood what is present. Actin is there, ATP binding site is there. Then we also learned about actin. Actin, it is globular actin, and F-actin, that is helically coiled. And we also understood who are the villains, that is regulatory proteins. You have troponin and tropomyosin. Actin and myosin are contractile protein. Troponin and tropomyosin are regulatory proteins. This question also has been asked, okay, one of the years. So this is the structure given in NCERT, and they keep asking the fill in the blanks, or like they ask, like you have to answer diagram based questions. Okay, then we understood neuromuscular junction. How exactly are we unmasking myosin binding site on actin? Okay, students, since I already spoke about it, I thought I will just make a note here about it. Okay, oops, actually knock and come, I got scared. Okay, contractile protein is going to be actin and myosin. Okay, actin and myosin. And regulatory proteins are going to be who? Okay, regulatory proteins are going to be troponin and tropomyosin. And students, this is only for actin, okay? Tropomyosin. Got it? Okay. Regulatory protein. Not Bosphorus. It's one of the person who help us with the studio setup, everything, okay? So I got scared suddenly. That's it. Okay, yes. Actin, myosin, troponin and tropomyosin, regulatory proteins and contractile proteins. Clear or not? Okay? Yes. Fine. Now, sliding filament theory is clear. Here, this also I have written. Here, this is enough for you to understand. Okay, this is what is sliding filament theory. Got it? Yes. Now, students, we will do some quiz. Okay. Ready? Put fire emojis in the chat. Then we are going to start with our quiz. Let's see who is going to be scoring high marks. Okay. Come on, yeah. Who is that 3 conjuice in the class who hasn't liked the video? So much hard work and no likes. And then you will be like, ma'am, why are you not taking class, ma'am? All drama, you people are. Okay, yes, ready? Chalo, let's see who's going to answer. The heavy meromyosin is composed of. This is also a PYQ question. So remember this and keep. It's a PYQ. It's asked in lot of mock papers. Okay. You see any mock papers from any place, you will see this one question. Globular head and short arm of the meromyosin. Globular head of the meromyosin only. The short arm and tail of meromyosin. The ATP binding site and tail of meromyosin. Let's see who is going to be the first one to answer. Quiz link, Illa Priya. Here only on the board you tell. Answer. Ikkada cheppali. Quiz ledu. A. Globular head and short arm of the meromyosin. Very, very good. Okay, so that means you people have got +4 in the chats. Okay, Emperor hasn't got any marks. Emperor, you told A and D also. Both you will not get marks. Okay, now next question. See, this is the question also. This is from your mock papers only. Okay, I've taken from mock paper. Okay, yes. Ma'am talking in Telugu. Yes, so I thought you people are going to now make a meme out of it. But yeah, I think I did a good job. Yes, tell A, B and C. This is what I told you. This is one PYQ itself. This is one PYQ. Yes, A, F-actin, B, troponin, C, tropomyosin. Okay, you people read, students, and tell the option. Okay. Who's the first person to tell? Come on, let's see. +4. Yes, all of you put your scores. Very good. Roshni, Madhumati, Meghna. See, my regular students knows the drill. They know that, okay, after I answer, I'm supposed to put my marks. Three. Okay, C is F-actin, very good. A is troponin, very good. B is tropomyosin, excellent. Excellent. +8. Okay, +8. Whoever got +8, please put it in the chat box. Next question. Which of the following about the globular head of a meromyosin protein is not true? Okay. Which of the following about the globular head of myosin protein is not true? It consists active sites for actin. It has an active ATPase. It projects outward. It is a part of the light meromyosin. Very easy question, NCERT based question. +8, +4, okay. Good job, good job. Okay, yes, this also answer and you'll get +12. Okay. Yes. And like the video if you are enjoying the class. That is the only thing that you can give me, okay, in return, nothing else. Right? Yes. Good job, good job. It is a part of the light meromyosin chain, which is absolutely wrong because globular head is a part of HMM, heavy meromyosin chain. Okay, this you have learned in my class today, so you should be able to answer this. Okay, excellent, +12 marks in your wallet. Okay, yes. Last question. In a contractile, that means last question from this topics, okay? In a contractile unit, each actin has, oh yes, this is also a PYQ. This is also a PYQ and this is also a mock paper question. I think I took it from one of the mock paper. I'm not sure, students. It's a mock paper question. Okay, yes. Yes. Come on. Tell me the answer for this. 2 F-actin polymer made up of F-actin monomer, 2 G-actin polymers made up of G-actin monomers, 2 F-actin polymers made up of G-actin monomers, and 2 G-actin polymers made up of F-actin monomers. So you should know monomers and polymers. Is there anyone who has answered? Come on, think and tell. It's easy only. Students, polymer you should know, monomer you should know. G-actin monomers or polymers? That much if you know the answer. Two F-actins are there that you can easily identify. Yes. I'm copying, you're cheating. Okay? You're changing people's name. Yes. 2 G-actin polymers made of F-actin monomers, sir. How is that right, students? How is that right? Think and tell me. 2 G-actins? Where did we tell 2 G-actins? 2 F-actins are there because both the strands, you saw, no? Like this 2 strands are there. So 2 F-actins is right, not 2 G-actins. So 2 F-actin polymers made up of G-actin monomers. This is the answer. C is the answer. Okay? Yes, some of you, I think one person has answered, but yeah. Okay? You cannot tell that as a polymer, no? That's why. Monomer or polymer, yeah. Got it? Me correct. Okay, very good. Okay, remember about 2 strands. You cannot tell, you can never say 2 G-actin polymers. What did I tell you? 2 F-actins are there. Okay, 2 F-actins are there, which are formed by many G-actins. Okay, so this is 2 F-actin polymers made of G-actin monomers. 2 F-actin polymers made up of, this is wrong. 2 G-actins, 2 G-actin polymers, no. Okay, so among these two, don't get confused because you never learned about 2 here. You learned about multiple G. Okay, multiple G-actins. Got it? So there's nothing to be confused. Maybe you people just got confused, like, reading and everyone were telling D option, no? So people got confused. Got it? So F-actin is the polymer, many polymer. Okay, so F-actin is a polymer made up of G-actin monomer. Clear? Okay, I'll add it here on the notes also so that you people are not confused. Wait here, no? Yeah. Okay, many monomers. Okay, wait. Okay, many ends up forming polymer of 2 F-actins. Okay, I've added it here so you people are not confused. Done. Now, students. Sorry. I'm neither at. Okay, now we'll take a cutie break. Will you all join back? If you're not going to join back, no break. If you don't take break, then you will not be able to do skeletal muscles. Okay. Yes. So for skeletal system, now what's the time? It's almost 8. Okay. Go have something and come. Okay. Since without skeletal system, you're not going to score marks. Okay, you're not going to score marks. So those who are not going to join back, your loss. Okay, so don't make yourself at loss, because I'm going to give you a lot of tricks to remember every single thing. So the only thing you have to do is just be in my class. Okay, yes, bye bye. Suvra, the moment I go out itself, it will become 10 minutes, dear. For me to drink one tea also, there won't be time, because I have to make everything here. That's why. Okay, that's why 15 minutes. Got it? So people also go have something. You can be here only. You can talk to each other. No, that's what you people do. So you can talk to each other, but make sure that the target we reach. Okay, let's put target. How much target? You all back? Yes, even I am back. Okay. How much are we on the likes? Ready? Okay, good job. Okay, now students, the first thing that is given is about your red and white fibers. Okay, now one more thing I wanted to tell you people is sliding filament theory is given by okay, Huckel and Huxley. Okay, Huxley and Huxley, both the people. Oh, that's it. That's not there, but then in case somewhere they see. Okay. Okay. Yes. Now see here, red fibers and white fibers. Okay, red fibers, from the terminology itself you understand it is red in color, right? So here you understand high myoglobin. Okay, pigment myoglobin. Here it will be low myoglobin, white or pale in color, red in color. Okay. Now higher number of mitochondria, here lesser mitochondria will be there. So here energy yielding will be more because powerhouse is who? Mitochondria. So higher number of mitochondria, here lesser number of mitochondria, can utilize large amounts of oxygen for ATP production. Large amounts of oxygen for ATP production. Clear? Cannot use large amount of oxygen, lesser ATP production. So opposite, just study one and you can write the opposite. Number of sarcoplasmic reticulum is lower. If sarcoplasmic reticulum is lower, means what? The amount of calcium yield also is going to be lower, right? But here if you see, sarcoplasmic reticulum is higher. You will understand why. See, slower rate of contraction for longer periods. Okay, slower rate of contraction. This is also linked to this, because if calcium is less or sarcoplasmic reticulum is less, that means the calcium is going to be less contraction. So here what will happen? Slower rate of contraction for longer periods. And here students, aerobic muscle fibers, here anaerobic muscle fibers. See, faster rate of contraction for shorter period. So if you want to say, let's say you want to run on a treadmill, okay, or you want to sprint, okay, very fast for 8 minutes or something, you will be using your white fibers. But let's say you want to run a long marathon, that time you will be using your red fibers. Understood? So that is how the difference is. Red fibers will be used for longer period of time, shorter energy but longer period of time. If you want to run a 15 kilometers or 5 kilometer long marathon, you will use red fibers. If you want to do a sprint or like a sudden something, like for a very shorter period of time, you will be using your white fibers. Is this clear? Yes or no? That's it, nothing much about it. Okay, chalo. Types of skeletal. So you have two types of skeletal system. You have appendicular and axial skeleton. Okay, so appendicular is your forearms, okay, and your legs, limbs. Okay, now axial means your skull. This portion, this middle portion is going to be here. So if you look here, you will see the blue portion is the appendicular. That means you have your girdles, you have your hands, legs. Okay, and here if you see, you have axial skeleton, you have your skull, your middle portion, right? Whatever is there, ribs, sternum, okay, vertebral column is going to be part of it. Now students, the most important thing that you have to know about your skeletal system is about your axial and appendicular skeleton. Axial skeleton has how many bones? Tell me in the chat box. Axial skeleton has around 80 bones. Now we have to count if we are writing it. Okay, when we are writing, please count if we are making up to 80 bones. Okay, 80 bones. Ready? Okay, yes. So we start with whom? Obviously the skull. Okay, skull. Now students, in the skull, you can count the skull bones and your facial bones. Okay, you can count your skull bones and facial bones. So let's say if I categorize the skull portion into two portions, where you have your skull bones, that is the part of the cranium, your cranium bones, and your facial bones. How much it will be? Facial bones. You have around 14 facial bones. Okay, and cranium bones you have 8 of them. Okay, please do the maths. Please do the maths. Be responsible. Okay. Yes. Now, other than the facial bones, you can also include your ear ossicles. You can also include the ear ossicles. That is 6. Okay, 3 each. So 6. You have 2 ears, no? 6. So this is the number. Okay, this is the number. Right? 22. And you have 6. Okay, 6 more, that is 28. Usually, hyoid bone is present at the base of the buccal cavity, and I wouldn't say we can consider it as a part of the skull. We consider it as alone. Okay, let's consider them alone. Consider them alone. Okay. I think I'm low on ATP. That's why my tongue is twisting. Okay. So yeah, when I talk for a longer time, my tongue starts twisting. So yes, hyoid is a bone which is also called the tongue bone, and it is present at the base of the buccal cavity. Okay, base of the buccal cavity. So students, this is clear. Cranium, facial bone, ear ossicles, and your hyoid. After you come down, see, after the hyoid bone, you come down, what do you see? You definitely see your vertebral column, but most importantly, you see your sternum. Okay, sternum. Sternum. Then you write vertebral column. Then what? Ma'am, rib cage or the ribs. Rib cage is all of them together. Ribs. Right? Did we miss something? Did we miss something? Skull, hyoid, ear ossicle. In the skull, you have cranium plus facial bones. Under the skull itself, I have written ear ossicles, but actually this whole portion, if you tell the whole thing, okay, ear ossicle, facial bones, cranium, hyoid, sternum, vertebral column, and ribs. Done? Sternum is 1. Okay? Okay, sternum is 1. Yes, very good. Vertebral column, we have to see the number. No, but we know the number, but still, we have to see the number. And then again, ribs. That also we know 12, but still, we will go one by one of each. Okay, we'll go one by one. Now, chalo, here only we'll write off. Okay. Now, student, let's go with first your cranium. Okay, cranium. Cranium is very, very important. So, the trick to remember cranium, all of you tell me in the chats. Yes, one flat bone is your sternum. But yes, tell me the way to remember your cranium bones. Ma'am, I know there is a trick. Okay, cranium is what? Brain box. Okay, so to remember the bones of your cranium, you have to remember this one trick which is called step off. Step off. Okay. That means S for sphenoid. It's not in order, but yeah, you will be able to remember. T for temporal. Okay. E for ethmoid. P for parietal. O for occipital. F for frontal. Now, remember one thing, out of this, 2 bones are going to be paired. So, I always told you to remember this way. PT teachers, PT teacher, you know, no? Physical education teacher. PT teacher will always come as a pair. One boy will be there, one girl will be there, because they will be coming to a co-head. So, there will be a girls, right? There will be boys. So, for that, we will have 2 PT teachers. One boy, one girl. So, always remember, PT teachers are going to be in pairs. So, only bones which are going to be paired among this, okay, or a present in pair is going to be your PT. P for parietal, P for parietal, T for temporal. Clear? PT teachers are always going to be in the form of a pair. So, these people are paired. Got it? These people are paired. So, the rest of them all are single people. So, how are you going to remember this? Step off. Okay. So, cranium bones, the way you're going to remember is step off. Okay. So, you will start with your frontal bone. Okay. Frontal, parietal, occipital. Okay. Now, remember, whenever we talk about actions, okay, to teach. Okay. So, frontal. Okay. They thought I'm sick. the team members thought I'm sick. That's why they're peeping and seeing what is wrong with this ma'am. Okay, before, if they would have seen all this, they would have thought that I'm trying to do something out of the world. Okay, yeah. So frontal, okay, frontal you have. Yes, ma'am, frontal I have. Then whom do you have? Parietal you have, temporal you have, occipital you have, ethmoid and sphenoid you cannot show. I cannot show you, okay? But remember, in that your sphenoid bone, okay, very important, okay, very important thing about your sphenoid bone is, students, this bone articulates with all other bone and they have a small depression called as sella turcica. Okay, they have a small depression called as sella turcica in which your pituitary gland is going to be there. Okay, pituitary gland is going to be there. Got it? So sphenoid is going to be the bone that articulates, okay, articulates with all the bones, all the cranium bones, okay, with all the bones. And students, what is that? They have a small depression, okay, like a small depression, we tell, no, like a furrow, which is called sella turcica, which I have taught you in chemical coordination, okay, sella turcica, in which your pituitary gland is going to be there. Okay, pituitary gland. Done? Yes. Exactly, that's what they asked me. Are you okay? Like this they asked. First that person came and saw me doing all this. Then the next person comes. Now tomorrow they'll tell in the group, Gopika Ma'am is doing a different kind of aerobics in the class. Okay, she's like twisting herself, then rotating her head. If she could rotate, she would rotate. Okay. So it is very sad, students, what and all I have to do. Got it? So yes, is this clear? So cranium, you have 8 bones. How will you remember? Step off. Okay, step off. If someone is eating your brain, what do you tell them? Step off. Don't you come near me. Like that only you think. Okay, cranium, brain box, step off. S for sphenoid, T for temporal, E for ethmoid, P for parietal, O for occipital, F for frontal. In that, who is the one who is going to be paired? PT. Okay, parietal and temporal. Parietal and temporal. Clear? Yes or no? Yes, that bat shape we are going to show. I'll show you that. Okay, bat shape I will show you very, very soon. P for parietal, O for occipital, F for frontal. Okay, my own. Done? Yes, clear? Fine. Moving forward. Now students, here is the diagram for you. So look here. You can clearly see the portions which I showed, right? Which is the blue color one? Ma'am, it is the frontal. All of you touch here and tell me, where do we check the fever? We check here. This is your frontal, right? This is your frontal. Then whom do you have? Parietal, okay? Then here, behind our ears, temporal. Then here, this portion we have occipital, which is going to have condyles, okay? Which is going to articulate with our vertebral column, okay? Now, what did I tell you? Sphenoid. Students, if you look at the structure of sphenoid, you will see it is kind of like a bat-shaped, butterfly-shaped, but more like a bat-shaped structure, which actually articulates with all the bone. If you see, it is articulating with frontal, ethmoid. Can you see? This is ethmoid. It's articulating with frontal, ethmoid. It's also articulating with your temporals. It's articulating with your occipitals. It's articulating with your parietal. So basically, this is the bone that articulates with all the bones, and it also has a small depression called as sella turcica, inside which the pituitary gland is going to be there. Done, done, done. Yes or no? So how are you going to remember it? Step off. Okay, so again go frontal. Okay, this you should know properly, students. Frontal, parietal, right behind your ears, temporal. Okay, and this portion, occipital. That occipital is going to have a condyle, okay, which is going to allow us to articulate with the vertebral column. Ethmoid is the only thing we can't show, but you can see ethmoid here. You can see ethmoid here. Yes, it looks like a bat, no? Exactly, very good. People are very smart. Okay, step off. In that step off, 2 people are going to be paired. Who is that? PT teachers. PT teachers are always going to come in pairs. This is this one, P, parietal. That's it, also parietal. So, this 2 portions. See, parietal, parietal, temporal, temporal. Okay, parietal, parietal, temporal, temporal. Done, done, done. No doubts, right? That's your cranium bones for you. 8 of them. Okay, now facial bones, students, you do not have to study the facial bones because there's only one diagram given in your NCERT which has only few portions of the facial bones. But otherwise, for your knowledge, remember facial bones are 14 of them. Okay, facial bones are how many? 14 of them. So here, if you see the diagram, I'll show you the diagram. You start with your nasal bone. Okay, nasal bone. Then obviously, you know, maxilla, mandible is there. Now, inside the nasal, you have your inferior nasal concha. Okay, inferior nasal. Okay. Then if you go here, all of you know, smile and you put blush, no, that is your cheekbone or your zygomatic bone. Okay. Then eyes, you have lacrimal bone. Then here you have single one bone called as the palatine bone, and then here you have a vomer bone which is single. Okay, vomer bone which is single. So again, nasal bone, maxilla, mandible, you people know. Zygomatic bone you can remember. Lacrimal bone you can remember. Right? All this you can remember. Yes. Okay. Perfect. Students, now I was doing all this. Okay. Now the security saw me doing it. So basically tomorrow my poster will be put on the office that this ma'am. Okay. So at this point now no more actions. Okay. We will only learn. So 14 of them. Clear? Yes. 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 Mandible, vomer is going to be single. You have your palatine. Okay. Vomer, that is only names, little tricky portion. Others are not tricky. Okay. Palatine and vomer. Other things are easier for you people to remember. Yes or no? Okay, no more actions will be done. It is problem to my yes. Done. Okay, yes, fine, this is done. So facial bones, I'm not going into the depth of it because if you go to my old videos, it is there, but then I think it is only few words that is given in your NCERT. Okay, so let's stick to NCERT because later students will be commenting, ma'am, why did you teach so much? We can't understand and all of that. So that's why we are skipping on that. Not skipping, I just told you the general thing. We are not going into the depth of it. Okay, now ear ossicles. Ear ossicles, students, is very easy to remember. What is the trick to remember ear ossicles? All of you tell me in the chat box. Miss has. Okay, Miss has. Okay, what does she have? You can write humor, you can write anger, you can write style. Okay, anything you can write about your teacher. Miss has what? Okay, Miss has what? Yes. Now, what disclaimer? Disclaimer, no people are there. Okay, now moving forward, Miss has. Okay, even if you don't write this, I'm okay with it. Just write Miss has also, no problem. Okay, if there are some, you know, like dictionary, Oxford dictionaries in the chat box, they'll be like, ma'am, Miss has, it's not even a sentence. What is it? For those people, you can write Miss has, humor, style, but the same words you have to use. Humor, anger, style, or humor, A for something, and style. Okay, so M for malleus, I for incus, S for stapes, which is the smallest bone. Okay, then H, student. So what is Miss has? Has is the shape and Miss is the name of the bones. Okay, so H for hammer shaped. Hammer shaped, which is hammer shaped? Malleus is hammer shaped. A is incus, anvil shaped. Okay, anvil shaped. S for stapes, stirrup shaped. This the names were given from the olden days tools. Okay, as you can see hammer, as you can see stirrup, as you can see anvil shape, sorry, anvil and stirrup shape. So students, these are the olden days tools that was used, so the names also come from there. Okay, so Miss has. M for malleus, I for incus, S for stapes, and hammer shaped, anvil shaped, and stirrup shaped. Clear or clear? Miss has, very easy to remember, no? So ear ossicles are going to be 3 each. Okay, 3 each. So you have 2 ears, so obviously 6 you are going to consider in number. Okay, now when we talk about hyoid bone, students, nothing much about hyoid bone. It's a single bone. Okay, single bone. This can also be called the tongue bone. Okay, tongue bone. And it is a U-shaped bone. And it is present at the base of the buccal cavity. Base of the buccal cavity. That's it. Okay, base of the buccal cavity. That's all you have to remember. Okay, that's all you have to remember. Got it? Yes, fine. So ear ossicles, Miss has. Okay, skull, that is cranium, step off. Okay, step off. Now, vertebral column, when we talk about, it's very easy to remember the whole thing, okay, because we have just a formula, no? So, what is the formula, students? You have to remember. So, first I think I will show you this. Okay, first I'll show you this and then we will come into this. See, look here. Can you see the different colors? Okay, these are the bones that are present. Okay, these are the bones that are present. Which are the bones that are present? Cervical, thoracic, lumbar, sacrum. Okay, this is sacral and coccygeal is fused to make the tailbone. To make the tailbone. So, tailbone. Okay, so CTLSC. CTLSC. Now, the formula stays C7, T12, L5, 1, 1, fused, fused. Or you can write 5, 4, fused, fused. That means both together will form one tailbone. Okay, one tailbone. So, tell me the formula, all of you. Tell me the formula, all of you. In the chats. Anyone who is not telling? Good job. C7, T12, L5, S5, coccygeal 4, but they are fused, so you put the bracket just like how you do it for your botany. Okay, how you do it for your botany. So just put the like fusion, and this will give 26 bones, and this is seen in adults. Okay, in adults. What about in infants? In infants, it's going to be C7, T12. These two are not going to be fused. Okay, L5, S5 and your C4. That makes a total of how many, students? 33 bones. This is going to be in infants. So all of you remember tailbone. Okay, so remember in males the tailbone is going to be more like this towards inside. Okay, more towards the inside. But in women it is going to be like this. Okay, it is going to be like this. Why? Because the women have to give birth. Okay, because the women has to give birth. This is the situation. Okay, situation. Clear? Yes or no? You can write 1 also, no problem. You can write 1 also here, no issue. You can write 1 also because these both are going to fuse. But the actual number, I have just written it here. And these two. Okay, these two together forms the tailbone. Okay, together forms the tailbone. Clear? Now, let's go back to understanding this with the help of this. Okay, now look here, students. See. Cervical vertebrae, 7 bones are there. In that you have to remember 2 bones. Okay, first is going to be atlas, then it is going to be the axis. Okay, first is going to be atlas. Now remember that the atlas is the one that is going to articulate with your occipital condyle. Okay, occipital condyle. What is condyle? The one that is going to help with the articulation. We need someone for articulation. So at the end of the occipital, you will see like this condyles. Okay, which is going to help in articulation. Articulation. So who is going to articulate? Atlas is going to articulate with occipital. Okay, then second one you have axis, then you can just write C3, C4, C5, C6, C7. Okay, then cervical vertebrae 3 to 7. Okay, then thoracic vertebrae, student, T1 to T12. Simple, T1 to T12. Okay, thoracic vertebrae you go from 1 to 12. Lumbar vertebrae, the strongest one, L1, L2, L3, L4, L5. Okay, then sacrum, 5 fused bones, here 4 fused bones. Sacrum and coccygeal together forms your tailbone. This is the tailbone. Can you see this tip only? I told you that it is going to be more deeper for the male, but like this for the females. Why? So that we have to, like the birth has to be given. Done. Now look at your atlas and axis, students. You will notice that one can get into another one like this. Articulation will be done like this. Okay, let's say this is your atlas. To that, axis will articulate like this. Understood? Yes, odontoid process. Okay, articulate like this. This is your vertebral column. That's all. Vertebral column is very easy. Actually, your entire skeletal part is just about your bones. You just have to learn the names of your bones. This is the diagram given in your NCERT. So, cervical vertebrae, thoracic vertebrae, lumbar vertebrae, invertebral disc so that we can do the bare minimum movement. Whatever movement we are able to do is because of the cartilage which is present between the vertebrates. Okay, sacrum and coccygeal. Now students, here you can see atlas. Can you see how axis is made? Axis is made in such a way so that they can articulate with each other. Okay, let's say this is your axis. Okay, this is the axis, this is the atlas. So to this, like this, articulation can happen. And remember, this atlas will articulate with your occipital. Below they will have condyles. Okay, everything will have condyles. So that means what? To allow the attachment, because they are all bones separately, you have to attach and make an entire vertebral column with the skull. So that will be done with the help of articulation. And this is your odontoid process. The two fingers which I showed you, know, this is your odontoid process. That means helping with the articulation. Yes, yes, atlas and axis, atlas and axis. Okay, next most important portion of your axial skeleton is sternum, who is a single bone. Again, single bone, but a very important bone. Why? Because this is the person who is going to help with the vertebral column, the formation of the rib cage, everything. The whole thing is happening because of this Mahan Vyakti. Okay, Mahan Vyakti. Yes. Dicondyle skull, you people studied in the neural nervous system also, no? Yes. Okay, students, this look like a dagger shape, but for me it looks more tie-like. Okay, dagger shaped. Okay, dagger shaped. Now remember, these each of the portion, no, they have names actually. Okay, this is one portion, this is one portion, and this is a small ending to this. Now remember, this is the prosternum. Okay, prosternum. Obviously this will be the mesosternum. Mesosternum. And this is your sphenoid process. Now students, these people, this entire sternum helps in the formation of the rib cage, as you can see here. Okay, as you can see here. Okay, you will see that how the vertebrae, how the entire ribs are attached to the thoracic vertebrae and in the ventral side to the sternum. Okay, the ones which attach are called the true ribs and the others are called as false rib, and then you have floating ribs. So sternum plays a very important role. So this portion is the prosternum, this is the mesosternum, and down you can see a sphenoid process. Clear? Okay, clear? That is your sternum for you. Now rib cage, very, very important. Now students, here what the question will be asked, the numbers. Okay, which is the true ribs, which is the false ribs. Now remember that I'm telling you are a true GMA. That means what? You will share my video, you like my video, whichever class I'm taking, you will join my class, right? There is some of students like that. Now it is very important that you remember that what are these true ribs. Now true ribs, students, true ribs are the ones who has two. Normally what a rib should do? Ventrally it should attach to the sternum, dorsally it should attach to the thoracic vertebrae. Now if you're not doing that, I will call you false, right? So same thing here, true ribs are going to be 1 to the 7th rib. They are going to be true ribs. Why so, ma'am? Because they are attached. They are attached to thoracic vertebrae plus sternum. Okay, this is ventral, this is dorsal. Correct? This is why I am calling them true ribs. Yes or no? Yes, 1 to 7 true, later false, later false. Very good. Yeah, you can call these true ribs one more word. What is that one more word? Vertebrosternal. Okay, vertebrosternal ribs. Because they are attached to the vertebral column and the sternum. So you can remember like that. Next is going to be false ribs. What these false ribs have done, students, they are so lazy that dorsally they are attached to the vertebrae. No problem with that, dorsally they are attached. But ventrally what they have done, instead of directly attaching to the sternum, they have gone and attached to the cartilage of the 7th rib. They have gone and attached to the cartilage of the 7th rib. Okay, clear or not? That is why these people are called false rib. Okay, so 8th 9th and 10th ribs, okay, are going to be called false ribs. And why are they called false ribs? Because they are attached to thoracic vertebrae. They are attached to the thoracic vertebrae dorsally and to hyaline cartilage, okay, or you can just write cartilage, and to the cartilage of the 7th rib. Okay, 7th rib. This is why we call them false rib. Correct? Or vertebrochondral ribs. Very good. Vertebrochondral. Okay, one second. Cartilages, different cartilages are there. Okay, like how you have hyaline cartilage. Okay, like that, different cartilages are there. Okay, ribs. Clear? Done? Yes or no? Yes. Now, moving forward again. Okay, next one is going to be, students, floating ribs. Floating ribs. Floating ribs also to the thorax, they are attached. That is, to the vertebrae, they are attached. But to the sternum, they are not attached. Not to any cartilage also. Not to any cartilage also. So, what they have done? Dorsally, they are attached. Okay, so this is going to be 11th and 12th. Again, dorsally attached, but ventrally free. Okay, they are not attached to anyone. They are not attached to any cartilage or anything. As you can see here, the 11th and the 12th. You will notice that these people are attached to nothing. They are just floating. Okay, these people are attached to nothing. They are just floating. Yes? Yes. Okay. Perfect. I hope no doubts. Yes. Vertebrochondral and vertebro ribs. Okay, floating ribs. Yes, these are vertebral ribs. Very good. Okay, these are vertebral ribs. This is the vertebrochondral because these are attached only to the vertebrae. So you can call it vertebro ribs. Okay, vertebral or vertebro. Okay, this is vertebrochondral and this is your vertebrosternal. This is your ribs. You can see 1 to 7 and how you can see how the 8th and 9th is attaching, 8th, 9th and 10th is attaching to the 7th, and you can see 11 and 12 not attaching to anything in the ventral side. Okay, next is going to be the bones of your forelimbs. Bones of your forelimbs. So for this, what is the trick, students? Trick. What is the trick? How are you, CM Pandu? Okay, one second, I have to turn like this. Yeah. How are you, CM Pandu? Normal question, no? You ask your friends, how are you? Same way, how are you, CM, Chief Minister Pandu? This is the trick to remember this. H for humerus. Okay. R for radius. U for ulna. C for carpals. M for metacarpals. And P for phalanges. How are you, CM Pandu? Very good. How are you, CM Pandu? How do I remember? This is my humerus. This is radius. This is ulna. This here. Or if you do this. This is carpals. They are arranged like, small pebbles, no? Like that they are arranged. 8 of them. Then this one is metacarpals. You can feel your metacarpals. Just touch and see. You can feel your metacarpals. 5 of them will be there. Okay. And then you go forward, students. Can you see the lines on my hands? Okay, like this. Can you see? Okay, now look here. 1, 2 on my thumbs. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. 14 phalanges. So, this lines, with that you can indicate the bones. Okay, got it? Yes or no? No, dear. Endomyosin is a covering, is a protective covering. Sarcolemma is the plasma membrane. Okay, plasma membrane. Yes, got it? Now, numbers is important. Obviously, humerus is going to be 1. Radius also is 1. Ulna also is 1. What did I tell you? Carpals are 8 of them. Okay, carpals 8, metacarpals 5, phalanges 14. A total of how many students? 30 bones. Okay, so your forelimb, one forelimb will have 30 bones, so that means both of them together 60 bones. Okay, 60 bones. And this is your appendicular skeleton. Okay, I don't have to specially tell, no, you understood, no? Appendicular skeleton. So axial skeleton up to vertebral column, rib cage we understood, and now we are doing the appendicular skeleton, the forearms and the limbs and the girdles. Okay, because we have to have this hands attached somewhere, no? For that we need girdles. So that is how we do. Okay, yes. So humerus, radius, ulna, carpals, metacarpals and phalanges. How are you, CM Padu? A total of 60 bones are here. Here also you can see same thing only is here. You can see how the attachment is there. Now your pectoral girdle is going to help with the attachment, which we will see slowly. Okay, is this clear? Now you move forward, you have the legs. Okay, legs. Now here students, I only made one trick. Okay, that is both the tricks I only made, but yeah, fly, but I think it might be difficult for you, some of you. Fly Tarzan, mountains, fly Tarzan, mountains and planets. Because it is GM production, my Tarzan can go out of the planet also, come inside the planet also. Okay, it is like my own production, no? I can just do anything and everything. So fly Tarzan, mountains and planet. F for femur, longest bone. T for tarsals, sorry, tibia. F for fibula. How you had radius and ulna. Okay, T for tarsals. How you had carpals. M for obviously metatarsals, and P for phalanges. So fly Tarzan, mountains and planets. Okay, yes. Six-fingered person is exceptional. Okay, most of the time it will be just the skin. Bone presence will be very less. Most of the time it will be just the skin. Okay, extra skin growth. Got it? So don't worry about that extra one finger. Now you think about the fingers that you have. Okay, count all the fingers. You people are there, no? When here in India something, okay, in India here fire is happening, you will think about Los Angeles and worry, oh, you in Los Angeles, if it rains, what will happen? Here it is burning down, no problem for that. All ulta-pulta system you people have. Now listen here. Fly Tarzan, mountains and planets. Okay. So F for femur, T for tibia, F for fibula, T for tarsals, M for metatarsals, P for planets, phalanges. Okay. Now here you see same thing is here, but the number is important, students. Obviously femur, one long bone, tibia, one bone, fibula, one bone, but tarsals are only 7 in number. Tarsals are only 7 in number, metatarsals are again 5 in number, phalanges are 14 in number. The only person who reduced is tarsals. Okay, the only person who reduced is tarsals. Clear? Yes, very good. Metatarsals 5 in number. Now, if you look at the number, students, definitely something is missing. That is when you will you will add your patella, okay, which is your knee. Can you see? Kneecap. Okay, kneecap. That makes a total of 30, right? Because this is 29 plus 1, 30. Again, your hind limbs are also going to be 60 bones. Okay, 60 bones. Clear? Forelimbs done, hind limbs done. Clear? So fly Tarzan, mountains and planet. How are you, CM Pandu? Okay, step off to remember the cranium bones, miss has to remember the ear ossicles, and then you don't have anything else to remember. Hyoid, sternum, rib, you know, 1 to 7 true, okay, 8, 9, 10th false, 11th and 12th floating. Okay, then you have your cervix, that is your cervical bone, thoracic bone, all that. So remember CTLSC. Okay, yes. Now we will learn about the girdles. Okay, girdles, and then we have some diseases, then we are done. Okay, are you all enjoying? Tell me in the chat box, are you all enjoying? Yes, perfect. Okay, did you all like the video? Okay, Yes, okay, good job. Now students, remember, we have to attach these portions. No, we have to attach the femur, we have to attach the humerus. So where is your humerus attached? All of you touch here and do this movement. Can you see? You can do this movement, right? That means your humerus is attached into a cavity, okay, and that cavity allows, like, there are joints, okay, like ball and socket joint, hinge joint, which is allowing you to do this movement, okay, movement. So here, remember, facial bones, you don't have it in your textbook. They have just mentioned few of them in your paragraph, not paragraph, in the image. So then no need trick for that, no, that's why. Okay, so here you see students, here you will see the humerus is attached to a cavity called as the glenoid cavity, and I always tell you people to remember the Barbie. All of you remember, we used to remove the Barbie's hand, like how we used to remove chicken, no, chicken leg. We used to remove the Barbie's hand and fit it in back. That time you will notice that the structure used to be something like this. Okay, you can't see, no, yeah. The structure used to be something like this, right? Barbie's hands, we had to fit it into one circle. So same way, the cavity also is like that, students. The bone, the cavity, and then you will be able to see processes also are there. But here you have to study your glenoid cavity and acromion process, okay, that we will come into. Now I'll show you lines of NCERT to get a better idea. See, look here. Pectoral and pelvic girdle bones help in articulation of your upper and lower limbs, okay, respectively with the axial skeleton. Each girdle is formed of two halves. You consider each portion as two halves. One half, together one full thing, okay. It's not like 1 plus 1, it's one half, okay. Now each half of the pectoral girdle, pec, upper, okay, pectoral, pelvic, okay. So when you see a skeleton in forensic, or when we see, okay, or like they dig and they found out that the skeleton is old, looking at the pectoral girdle, they'll be able to tell if it's a male or a female, because the pectoral girdle will be wider for the female, again because of the purpose of birth, okay, to give birth. So pectoral girdle is going to be more wider than that of the male. So that is an identification. I think in movies and all you would have heard it. Each half of the pectoral girdle consists of a clavicle, okay, clavicle. All of you touch here and tell me. Touch here. Can you feel a collarbone? No, don't tell ma'am, I'm very chubby, I cannot see the collarbone. Don't tell all that, just touch like this and tell. Last time I told, my God, they had a whole discussion about body positivity, okay. They're like, I am very chubby, I eat this food, I eat this food, I eat junk food. So that's why I'm scared to tell all these things. So here, touch here and see, you will feel the collarbone. That is your clavicle, okay, clavicle. And you will have a scapula, which is a triangular bone, okay, a triangular bone. Now, scapula is a large triangular flat bone situated in the dorsal part of the thorax, this portion, okay. This portion you will feel your, I think, okay, this portion you will feel your clavicle, right, and here you will feel your collarbone. Got it? So clavicle is to the dorsal part of the thorax between the second and the seventh rib, okay. Second and the seventh rib means till here it is extending, okay, this to this portion it is extending, okay. The dorsal flat triangular body. So here if you see, can you see how long it is extending? From the second to the seventh, okay. So all this triangular bone, and this is going to be your clavicle. This is the clavicle. Again, here's the clavicle and this is your scapula. The dorsal flat triangular body of the scapula has a slightly elevated ridge called the spine, okay, called the spine. Can you feel the spine? Here somewhere, can you feel the spine? A small sharp, okay, ridge, okay, spine, which projects as a flat expanded process. Now, which is the first process that you are going to study about? Acromion process, okay, which projects as a flat expanded process called the acromion. The clavicle articulates with this. The clavicle articulates with this, okay. Got it, all of you? Yes or no? Easy? So first attachment that you're going to talk about is the scapula, which is a triangular bone, and you have a clavicle, which is a flat bone, okay, like this, the collarbone. Now this clavicle has to attach to the scapula. For that, the first process you have is acromion process, okay, acromion process. So acromion process is where the clavicle articulates, okay, articulates with it. This below the acromion is a depression. So I told you in the Barbie thing, have you seen, they also give a depression like this. To that depression only you fit in this hands. So same way the humerus will be fit into a depression called the glenoid cavity, which articulates with the head of the humerus, clear, to form the shoulder joint. This is your shoulder joint. All of you can do all of this? Yes? Yes, ma'am. Each clavicle is a long, slender bone with two curvatures. This bone is commonly called the collar bone. Nothing is there to remember. You have pectoral girdle, two halves are there, one clavicle, one scapula, one clavicle, one scapula. Clavicle is attached to the scapula and the spine with the help of an acromion process, and your humerus is attached to a cavity, a small depression called the glenoid cavity. Ho gaya. Done, done, done. Okay, done, done, done. Yes or no? That's it. There's nothing difficult about this. 1 second, students. Okay. Yes. Okay. Good job. Okay. Next one. Your pelvic girdle. Okay. Pelvic girdle also, again, students, both the girdles are halves. Girdle meaning itself is where we, like a loop, we tell, no, like that only girdles are connections of two halves. So this side one half, this side one half. For this you have a trick to remember. Okay, you have a trick to remember. The trick is I play in class. What is the trick? I play in I play in class. This is the trick. I play in class. So easy to remember, no? So I for ischium, P for pubis, I for ilium, and C for coxal bone. Coxal bone. Okay. So remember, I play in class. Again, pectoral was divided into clavicle and scapula. Here also pelvic also has two halves. Okay. Which are the bones they have? Ilium, ischium, and pubis. Okay, ilium, ischium, and pubis. So I play in class. Ilium, pubis, ischium. These three together fuse to form your coxal bone. Okay, so look here, students. Each coxal bone is formed by the fusion of three bones. I play in class, last class. Okay, fusion of three bones, ilium, ischium, and pubis. At the point of fusion of the above bones, again, is a cavity called as the acetabulum, to which who will articulate? Your longest bone, femur, will articulate. Okay, which is your thigh bone articulates. The two halves of the pelvic girdle meet ventrally to form a pubic, okay, symphysis containing fibrous cartilage. Yes? Yes, I play in class. So, ilium, pubis, ischium together forms the coxal bone. Okay, together forms the coxal bone. That is the half of your pelvic girdle. Done? This is clear? This is clear? And in between you will have your sacrum and coccygeal forming your tailbone. Okay, and this is your tailbone, which will be more inside for the male and little outwards to the female. Any doubts in this, students? Now joints. From joints, what questions you will get? You will get examples. They will ask you the examples, okay, of joints. Suddenly I think everyone ran for dinner. These people know no sacrifice, nothing. Okay, fibrous joints do not allow any movement. So basically, students, three joints you have to know. One which will not at all allow any movement, one which will allow slight movement, one which will allow more free movements. Okay, so fibrous joint do not allow any movement. This type of joint is shown by the skull. On the skull, have you seen sutures? Okay, on the skull, have you people seen sutures like this? Like this one portion, like this one portion, like the sutures you have seen, no? You cannot move your sutures. So that is a type of fibrous joint. Okay, that is the type of fibrous joint. So by a flat skull bone which fuses end to end with the help of a, okay, end to end with the help of a dense fibrous connective tissue. Fibrous joint, fibrous you can think of tissue in the form of sutures to form the cranium. Sutures looks like, you know, like 2-3 pieces have been cemented together and there is a crack in between, like exactly like that sutures will look like. Okay, sutures. I think in this the emoji that you sent, it is not there. In cartilaginous joint, this will allow slight movement. So three types of joint: fibrous joint, which does not allow any movement. Example, you have to remember sutures in the skull, okay, or the cranium. Now next is the one that will allow slight movement. Okay, now in cartilaginous joint, the bones involved are joined together with the help of cartilage, like in your vertebral column. the joint between the adjacent vertebrae in the vertebral column is a pattern, or it permits limited movement. Okay, limited movement. There will be one set of people, like one set of parents who is very strict. Then there will be one set of parents who are not too strict, not too chill. Then there will be one set of parents who are very chill. Just like that only. No movement, some movement or limited movement, and compared to this, more movement. Okay. Now, here, see sutures. Can you see sutures? Yes, ma'am. Can you see vertebral, that is cartilaginous joints? Yes. And these are your cartilage. This is the reason why you are able to bend and pick something. Okay. Now, synovial joint only, students, little more is there. Okay. Now, synovial joints are characterized by the presence of fluid-filled synovial fluid. Okay, synovial cavity. That means there is a fluid called the synovial fluid, which is because of which we are able to do this ball and socket. Okay, knee joint, you can stretch knee, right? Yes, between the articulating surface of two bones. Such an arrangement always allows considerable movements. It's not like, oh, you can flip like this and bring this hand backside. That and all you can't do. There are people who do that in the dance and all. I've seen that India's Got Talent, you know, this twisting the whole thing and all of that. Okay, but that's not possible, students. These joints, I mean, for us it's not possible. These joints help in locomotion and many other movements. First one, ball and socket between humerus and your pectoral girdle. Think of this way, this, and even in your pelvic also. Pelvic also you can do, no? So, ball and socket. Second one, hinge joint. Hinge joint is your knee joint. Here, hinge. Door has no hinge. You pin, the door has a hinge and you'll open the door, no? Same way, knee also. Knee joint, okay? Yes. Now, pivot joint. Very, very important question has come in the previous years. Match the following has come for all these, okay? These are PYQs, so please write and study if you're not able to memorize. I try to make a trick, but it is too difficult to make a trick because if I put the first letters, you will not understand. So I have to make a trick like a whole sentence. Then you will be like, ma'am, to remember that trick, please give me another trick. So why to complicate life, students? So we'll keep it easy. Pivot joint between atlas and axis. Atlas and axis. Pivot joint. Okay. Gliding joint between carpals. Gliding. Between them also there is a thing, gliding joints. Okay. Then saddle joint, students, between carpals and metacarpals. Can you see between carpals and metacarpals? Metacarpals, carpals. Okay. This kind of a joint. Yeah, hinge is like opening and closing, even door. Okay, so saddle, students, very sad joint is who? Between carpals and metacarpals, because they can hardly make much movements. Okay, so saddle joint, sad joint. Okay, so think like this, saddle is between two people. When two people are together only, they'll be sad or happy. Saddle joint is between carpals and metacarpals, because why I'll tell you this, gliding and saddle joint, many students told me when I was teaching in class also, like batch also, they told me, ma'am, it's very difficult. We get confused between these two. Think like this, sad. You only become sad when someone is there with you or someone is not there with you. So saddle joint is between carpal and metacarpal. Carpal, metacarpal. Gliding joint is only between the, you know, carpals. Clear? Only between the carpals. So revise once again, ball and socket joint, pectoral. Okay, pectoral girdle and humerus. Then hinge joint, knee joint. Hinge joint, like you're opening the door, closing the door, like that, hinge joint. Then you have what? Gliding, carpals. Okay, then atlas and axis, pivot. Okay, saddle, between carpal and metacarpal. Done? It's actually easier when we learn it together. Okay, we repeat it, we learn it. So this is the joint cavity containing the synovial fluid, because of which we are able to do some movement. Okay, we are able to do some movement, and this is the synovial membrane. Clear? Cartilage is giving limited movement. Synovial is giving comparatively considerable movements, and your fibrous joint, no movement at all. Sutures, vertebral column, cartilaginous between the vertebral column, and you have synovial, ball and socket, pivot, saddle, hinge, and your gliding. Okay, glide is like slide. Yeah, exactly. Now students, last one, your diseases. Okay, because of this, what will happen, right? So myasthenia gravis and muscular dystrophy, multiple times asked question. Myasthenia gravis is an autoimmune disorder. Autoimmune means what? Auto means self mode, right? If you take auto mode in your phone also, phone itself will regulate the contrast, brightness, everything. Similar to that only, autoimmune disease means when our body starts hurting our own cells. It's not able to understand which is the foreign particle, which is my own cell. That is called an autoimmune disorder. Okay. So myasthenia gravis is an autoimmune disorder affecting the neuromuscular junctions. Neuromuscular junction is where we studied about your actin and myosin. Okay, actin and myosin. Clear? Thank you. Yes. So autoimmune disorder affecting neuromuscular junction leading to fatigue, tiredness, weakening, okay, and paralysis of the skeletal muscles, because you know how important neuromuscular junctions are. Then otherwise myosin binding site won't be visible. Otherwise also excitability should be there, right? For any movement to happen, neuromuscular junction should be extremely active. So this autoimmune disorder will lead to paralysis. So students, gravis, myasthenia gravis, think of graveyard, burying someone, no, like that only you think of. Autoimmune, killing the own cells, okay. Graveyard, think of killing to own cells, okay. Myasthenia gravis, because again students get lot confusion with muscular dystrophy and myasthenia gravis. Next is muscular dystrophy. Here, progressive degeneration of skeletal muscle, muscular dystrophy. So here, progressive degeneration of skeletal muscle, mostly due to a genetic disorder. So this is a genetic disorder, this is an autoimmune disorder. Yes? Yes, okay? Yes. Tetany, rapid spasm. Now let's say I did this, okay, or I did this. My muscles have contracted. Now you can see no contraction has happened, but I'm not able to relax. I'm not able to easily relax. That is because of due to low calcium in the body fluids. So when our body fluid calcium is less, what exactly happens? Contraction will fail, like contraction, and the entire process that you studied will fail, because only if calcium is released, the contraction, the sliding filament theory, everything can happen. If there is low concentration, after contraction, rapid spasm happens, then relaxation will not happen easily. Okay, so that is called as rapid spasm or wild contraction in the muscle due to low calciums in the body fluids. Then arthritis, all of you know, no, inflammation of joints, which we see nowadays in our parents, okay, in our grandparents. After one age, what happens? The joints, okay, between the joints, the fluid will be there, no, that will reduce, and slowly the joints will start, you know, coming closer. Okay, because of that, when they touch or do something, it will hurt. It will hurt very, you know, intensively. So inflammation of the joints, swelling, a slight swelling or redness. Osteoporosis, students, age-related again disorder. What happens? Because of the amount of estrogen less, especially in females, you will notice after the age of 40-45, our bone mass reduces, because of which by a small, let's say I just dashed here, immediately you will notice that there is a fracture. You will see, you know, grandparents also, they're just walking, they touch somewhere, immediately the bones will break. Okay, that is because the bone mass has reduced. Okay, bone mass has reduced. So age related disorder characterized by decreased bone mass and increased chances of fractures. Okay, decreased levels of estrogen. So estrogen plays a very important role in this bone mass. Okay, bone mass. So that's why females are recommended after a certain age to do you know, exercise, practice really well, make sure that their body does not become really weak, because as the estrogen content reduces, the bones mass also starts reducing. Okay, yes. Now gout is inflammation of joints due to accumulation of uric acid. And students, this is extremely painful, because when something, let's say you know, if I ask you to kneel down on salt, how do you feel? It will be pricking, no, every small bit will be pricking. Okay, that pricking pain you will have when the person is experiencing gout, because what will happen, the inflammation is due to the accumulation of uric acid. Okay, uric acid that to crystals, so that pain will be there. Clear? Principles of inheritance is done by Baswa sir, dear. Okay, yes, those who haven't liked the video, quickly like the video. Come on, I know some of you haven't. Okay, yes. Now students, answer this. Which of the following is present between the adjacent bones of the vertebral column? Easy question. Which is present between the adjacent bones of the vertebral column? Okay, so chapter is over. We completed up to diseases, and those who joined right now, I'll tell you that the notes will be shared on the Telegram channel here, Biology by Gopika. You can join. All the handwritten notes are there. So as you can see here, see, everything is written for you, drawn for you. Okay. So here everything, all the diagrams are there. Those who joined right now, you'll be tense. No, ma'am, what did you do in the before? We were in there. So all the diagrams are there. So quickly join this Telegram channel and take the notes. And also it will be present in the description box after the class. Okay, so by tomorrow morning, the description box will be updated and the notes will be available in the description box as well. So you don't have to go searching anywhere. Just open and start studying. Yes, what's the answer? Very good. Answer is cartilage. Okay, between the vertebral column, what do we have? Cartilage, because of which we have some limited movements. Assertion and reasoning. Osteoporosis is characterized by decreased bone mass and increased chances of fracture. Now, reason, common cause of osteoporosis is increased levels of estrogen. Yes, next question. So last time all of you got up to 16 marks. Some of you got, most of you landed up in 12 marks. So with 12 you can add now +4 for this. Okay, +4 for this. So now you are at 16. Okay, now you are at 16. Those who got 16, that is I think only Madhumita, will get 20. Okay, yes, answer for this, students. Okay, very good. What is the answer? Oh, sorry. Yeah, the answer is, this is correct, but this is wrong. So A is correct, R is not correct. Option number 4. Okay, so option number 4, that means right now you're at 24 and others are at 20. Okay, so those students who still wants to take the NEET Express, you can still join for the NEET Express. Also, you can avail for All India Test Series. Here is All India Test Series. You will get it at home at this price. I mean at combo. Okay, NEET Express 2.0 combo. And if you want hard copies of NEET Crash Book, okay, NEET Crash Book, you will get it at this. That is NEET Express 2.0 Pro. Okay, many students have taken Pro as well as many of them have taken combo, because you will be able to access to all the lectures that is present in the Wave. At the same time, you will be able to access to many ebooks, many mock papers. Every Sunday, you will have tests from 2 to 5 p.m. Also, for that, whatever portions has been completed. Okay. So, all the lectures, you will get unlimited access to Doubt App also you will get. Finally, got it? Clear? So, if you want to take this, you still have time. You're not too late, because right now only we got to know about the, the pattern change. So, I think NTA will get more on their toes, telling that okay, we need to make sure that everything happens well in this exam. So there's a high chance that only the hardworking students will go forward, and which is also the best thing, right? Because when hard work pays off, you feel like, yes, I have done my best and I got what I want. But sometimes when money and other things play, you cannot do anything about it. You feel actually very guilty and you feel like, why am I poor, or why am I like this, why am I like this? So you question yourself. But when it is hard work, it is like, okay, I can do my thing. Okay, I can do it and I will get the marks for it. So students, let hard work always pay off. Okay, because I know how hardworking my students are, and I have I know you people, even though it's online, I just know your names and I know how active you people are. So thank you so much for joining. I hope all of you enjoyed the class. The video has to reach many students only if you like and comment on the video. Let's say students, 10 comments, okay, are there in the video. The next student, the 11th student, when he opens, he'll be like, okay, 10 students have commented so positive. Automatically, the student will have a trust to open. So how well I teach totally will only be worthwhile when you people like and share the video and also comment. Okay, so take 5 minutes. See, for me to make this PPT, it takes at least 2 to 3 hours. It will take you only 2 minutes to comment. Just put whatever it is, your feedback, your criticism, you can put it in a nice way. Okay, so thank you so much, students. I hope all of you enjoyed all the tricks and also the GIFs that I used. I wanted to do 3D, but then something happened. It did not work at all. So yeah, let's hope and try next time it happens. All the notes will be available in the description box, also on the Telegram channel Biology by Gopika, and you can also follow me on Instagram, Gopika Biology, and also follow us on Vedantu NEET English Telegram channel, where you will get all the notes of PCB. So till then, thank you. Bye bye.